Since recording just started, I'll repeat. I'm Almazroua. I'm a fourth-year medical student, and for today's session, inshallah, I'll be explaining cell cycle and gametogenesis. These are objectives, and we're going to start by talking about embryology in general. Now, whenever someone says the word embryology, we can break it down into two simpler terms. We have the word embryo, which basically means an unborn baby or a baby in the uterus. And we have ology, which means the study of. So embryology overall means the study of unborn babies. And embryology can be a very interesting topic once you understand it really well. I know in the beginning, in foundation especially, it can seem very hard, but trust me, it's very easy once you start to grasp the concepts, and it can be very interesting as well. So can someone tell me what is the basic units of a human? Like, what are we made of as humans? Cells, exactly. So all of you said cells, and that's true. So in embryology in particular, cells are very important because we're making up a human. So literally, a human is being made. And human is made of cells. So we're going to need a lot of cells in the process of embryology, or like making an embryo, because cells after cells after cells end up being a human. So there's a lot of cell division that's ongoing, especially when we were like fetuses or embryos. So a cell cycle is the series of stages the cell goes through to divide and produce daughter cells. And this is something you know from high school. A cell divides and produces daughter cells. Now, in the cell cycle, we have two main phases. The first one is called the interphase, which is this phase here, from G1, S, and G2 phase. All of these fall under the interphase. And this is where the cell spends most of its time. We're going to talk about it more in details in the upcoming slides. And the other part of the cell cycle is what we call the M phase. Now, the M phase depends on the type of cells. Now, in the context of cell cycle, the type cells in our body are divided into two major cells. We have the somatic cells, which are basically all the cells in our body. Epithelial cells, hair cells, intestinal cells, all of these cells are called somatic cells. And then we have the germ cells or the sex cells. These are the ones that divide by a process called meiosis. Whereas the somatic cells, they divide by what's known as mitosis. And we're going to talk about them both in details in the upcoming slides, inshallah. Now, the interphase. So we said we have G1, S phase, and G2 phase. Now, the G1 phase is the longest out of the interphase. It lasts for several days or weeks. The cells differentiate and perform their specialized function. And it's just common sense. There is not much to explain here. In the S phase, it lasts for 10 to 12 hours, and the nuclear DNA is replicated in the S phase. Now, I've highlighted this in red because this is very important. Because I want you to understand this before we go into the cell cycle in more details, that the cells replicate their DNA or nuclear material before it goes into the division. So in order for us to divide the cells, we need double the number of DNA material or chromosomes. So this happens in the S phase before going into meiosis or mitosis. And the interphase, particularly the S phase, this is where the nuclear DNA is replicated. And the S comes actually from the word synthesis because we're synthesizing DNA. And then the G2 phase is relatively short. It's the shortest. And here the cells start preparing for division, which makes total sense because it's the last phase of the interphase and it's just before starting our M phase or meiosis and mitosis, depending on the cell. Okay, now we're going to move to the cell types in our body. So there are cells that are continuously dividing, which basically are most of the cells in our body, like epithelial cells, for example. When you have a wound or a cut, what makes your wound heal is basically your cells dividing, because we have a stem cell layer in our epithelium, and this stem cell layer keeps dividing. And as it's dividing, basically, it makes your wounds heal, for example. So these cells fall under the continuously dividing cells, and these cells are just continuously going over the cell cycles again and again. Now, the other type of cells are the terminally differentiated cells. Now, these cells go through the cell cycle once, and after they go through the cell cycle, they go and rest in a stage that's called G0. This one here I'm pointing out. Now, when they're at the G0, they can never leave. Now, an example for this would be the neurons. So the neurons in our brain just divide once, and then they rest at the G0 phase, and they never go back to the cell cycle. Once they're in G0, they stay there forever. And then we have another type of cells that are called facultative dividers. Now, these cells initially would go and rest in G0, just like the terminally differentiated cells. However, under specific stimuli, they can go out of the G0 and go back to the cell cycle. An example for that would be the liver cells. Now, I don't know if you've heard of that, but the liver is capable of like growing again. Like this is why we can donate parts of our liver. This is why liver donation is a thing, because you can cut a part of the liver and then the liver, subhanAllah, it just grows again. And this is because the cells in the liver are facultative dividers. So they're initially resting, but then when someone like resects a part of the liver, the cells can go back to the cell cycle and then divide again and produce more cells so the liver can grow again. So these are the different cell types in our body. Now the checkpoints in the cell cycle. Now the cell division is not a process that happens randomly. There are so many regulations that happen throughout the cell cycles, and these regulations are called checkpoints. What it means, it's literally checking if there's any error in the process of like cell division and the process of replication of DNA. It checks a lot of things. And if there is an error, these checkpoints either tell the cell you'll go back and correct whatever error there is if you can correct it. If you can't, then the cell undergoes apoptosis. And this protects us from a lot of diseases and a lot of syndromes that can arise from errors in like DNA replication or cell division itself. And we have three major checkpoints in the cell cycle. There is a G1 checkpoint, G2 checkpoint, and metaphase checkpoint. Your lecture didn't go deeper, so just know the names and where the checkpoints fall into. Now, we're going to talk about meiosis and mitosis. Now, the major difference between them is that mitosis occurs in the somatic or body cells, which basically are all the cells in our body, except the germ cells or the sex cells. Now, the meiosis is the opposite. It occurs in the sex cells, meaning the sperms and males and the eggs in females. Now, mitosis produces two genetically identical diploid cells. So the parent cell would divide and give rise to two daughter cells, and they're completely identical to the parent cell. In meiosis, however, it produces four, not two, and they're genetically diverse haploid cells, meaning they're not identical to the parent cells. They're different. And we're going to explain why later on. Mitosis involves one round of cell division, so we only have mitosis. However, meiosis, it has two rounds. There's meiosis 1 and then meiosis 2. I believe most of you already know this from your high school, but it's good to have kind of a major point before we go deeper into the cell divisions. Now, one important thing that will make explaining the cell cycle way easier is understanding the structure of a chromosome. Now, probably all of you know that this is how a chromosome looks like, but I want to explain it in more details in a context that helps you understand the cell cycle. Now, the chromosome has a short arm, which is this one, and a long arm. And in the chromosome, we can see that here is what we call a chromatid, and the other one is another chromatid. These two sister chromatids are joined together by a centromere in the center. Now, why is that important? One thing I want you to remember here is that this centromere is what decides the number of chromosomes for us. And say, for example, we had this structure here, and we had a centromere in the middle, and we had this structure here, and we had a centromere in the middle, and we had this like very weird looking structure. Suppose it's a chromosome, and we had a centromere in the middle. Can someone tell me how many chromosomes did I just draw here? If we said, the centromeres decide the number of chromosomes for us, can someone tell me how many chromosomes do we have here?
3.
Yes, exactly. There are 3 chromosomes. Now, this concept is important because when we go into details in the cell cycle, you will understand that no matter how many chromatids you have, whenever we say the word chromosomes, it means how many centromeres do we have. Now, the process of studying chromosomes is called karyotyping. It's basically chromosomal analysis. Now, this is used in the hospitals to detect many syndromes. Say, for example, Down syndrome is called trisomy 21, which means there are 3 chromosomes instead of 2 at chromosome number 21. So when you look at the karyotyping in Down syndrome, for example, I'm just giving you examples so you understand what karyotyping is, they would see an extra chromosome here. So this is how they would confirm, okay, this person has Down syndrome. Other than that, karyotyping can also detect if there's any abnormalities, not only in the number, but also in the structure itself. Now, if you look at the karyotyping here, we have 46 chromosomes and 23 pairs. And why did I say 46 chromosomes? Because if you see here, if we zoom in, they're not connected by centromere. Each chromosome has its own centromere here in orange, and they're not connected together. So this is a chromosome and this is a chromosome. They are a pair of homologous chromosomes. When I say homologous chromosomes, meaning chromosomes that basically they have the same structure or a very similar structure, but they have different alleles. So one is inherited from the mother, If you know what karyotyping is, I believe that's enough at your level. Now, there is a checkpoint. Does anyone have any questions about anything I've explained so far? All good. Okay, perfect. Now we're going to dig deeper into the process of mitosis. Now, mitosis, as we mentioned earlier, it ensures equal distribution of chromosomes into two daughter cells. So the end result of mitosis would be two daughter cells that are identical to the parent cell. Now, we have 4 key stages in mitosis. And I remember them by saying like PMAT, which are prophase, metaphase, anaphase, and telophase. Now, before the onset of mitosis, there occurs the synthesis of new DNA, which, as we mentioned earlier, it occurs at the S phase of the interphase. So it's before mitosis, we've already replicated the DNA, and now instead of having 46 chromosomes or 46 genetic material, actually, we'd have double that number, which would be 92. I've copied this word by word from your slide because I thought it might be confusing to some of you. And basically, the genetic material becomes tetraploid, but the number of chromosomes remains diploid. Now, you might be wondering, what does that mean? We've replicated the DNA, then why aren't the chromosomes 92? Why are they 46? Basically, what happens is, so I'm just going to draw it to make it easier, yani. So we have one cell, and we have 46 chromosomes. Now, let's say each one of these I'm drawing is a chromosome on its own with its own centromere. So chromosome, chromosome, chromosome. Let's assume I've drew like just 46 chromosomes. What happens during the S phase or the DNA replication is that we would have double the number of these chromatids. So basically, we said the chromosome is made of chromatids. Now we have one chromatid and a centromere in the center. So this is a chromosome because we have one centromere. What happens in the S phase is that we would have 2 chromatids instead of 1 attached to a centromere. So this is 1, 2, 3. And let's assume I drew 46 of this, okay? So now what do we have? I'm just going to change the color. Here we have 46 chromosomes and 46 chromatids, right? Because we have one centromere and one chromatid in the center. When we replicate the DNA, we'd have double the chromatids, which makes up 92 DNA or genetic material. This is what it's trying to say. The DNA material is doubled, becoming tetraploid, meaning 92. However, the chromosomes remain diploid in 46 because, as I mentioned earlier, the chromosomes are counted by the number of centromeres. And here we said, let's assume they're 46. We have 46 centromeres, meaning we have 46 chromosomes. However, how many chromatids do we have? Double the number. It's 92. Does that make sense to all of you? I know this might be confusing, so I can repeat if someone didn't understand. Did you get this point? Because we're going to go over it again in meiosis, so I want you to understand it from the beginning so it's easier. Yes. Okay, perfect. Since you understood. So we agreed and we count the centromeres, and this tells us how many chromosomes we have. And for this reason, we have 46 chromosomes. However, the genetic material is tetraploid, meaning we have 92 because we have double the number of chromatids, not the chromosomes. Okay. Now we're going to start by talking about prophase. Before getting into what happens in prophase exactly, I just want to tell you how the state of a cell that's not dividing is. So in a cell that's not dividing, the genetic material or the DNA in the cell is very loose. It's not condensed. It's just swimming within the nucleus. So it's very loose and just all over the place. Now, when you want to divide the cell, we can't have that because, as I mentioned earlier, cell division is a very precise process. It's very regulated. So if the genetic material is all over the place, it's very hard for the body to kind of keep it as regulated as it's expected to be. So what happens in prophase, since it's the first step of cell division, is that the chromosomes start to coil, contract, and condense. Basically, they start getting so dense that they don't move and they're not as loose as they were before. This helps us, or helps the cells, regulate themselves in the process of cell division. So you can see what's happening. So you can regulate it. Now, the chromosomes become shorter and thicker, which makes sense. Anything that's condensed is kind of shorter and thicker. It just makes sense. It's condensing together. But they remain initially, in the initial phases of prophase, they remain invisible under a light microscope. Now, this is something I've mentioned earlier, that the chromosome 2 parallel subunits, which we call the chromatids, and these chromatids are connected by a centromere or the kinetochore. It's just another name. It's just the name of a protein. Near the centromere is what we call a kinetochore. This is just a terminology for you to remember. Now, initially, again, the nucleus remains intact and the nuclear membrane does not integrate. Does not disintegrate. Okay. Now, these are the changes that are happening mainly in the nucleus. We're going to move to the changes that are happening in the cytoplasm and prophase. So basically, in the cytoplasm, we have a structure that is known as the centriole, as shown here. What happens in prophase is that they duplicate. So instead of having one centriole, we have two now. And that's very important because we need each centriole to go to opposite poles of the cell. So this is a cell at prophase. So a centriole would go here, and another centriole would go here in opposite poles. Now, why is that important? Because these centrioles, a very important structure starts to arise from them, which we call basically the microtubules or the mitotic spindles. Now, these microtubules would start to radiate like that, in this way, from the centrioles. Another structure that keeps the centrioles attached to the poles, because we really need them to be in the poles to be able to divide this, the chromosomes and to undergo the cell division, is what we call the asters or the astral rays. Now, these astral rays, let me just draw them in a different color, are present in the opposite side. So these green structures here. So they matter because they keep the centrioles attached to the opposite poles so that, again, it all goes back to the same point. We want to keep the cell cycle as regulated as possible. So the asters or the astral rays are responsible for keeping the centrioles attached to the poles of the cells, the opposite poles. Now together, this is just another, again, terminology, the spindles with the asters, they form what is known as the achromatic figure, also known as disaster or amphiaster. Okay, this is just terminologies for you to remember. So this whole structure here, the mitotic spindles and the asters, they're known together as achromatic figure. I know they're overwhelming terminology sometimes, and no, there are a lot of names to remember, but when you, inshallah, hear them repetitively, they get easier. Okay, now the late prophase is also known as prometaphase. You will not see prometaphase in all the resources. However, it is mentioned in your lecture, so I'm going to mention it. So prometaphase is late prophase and just before the beginning of metaphase. So you're going to see in some videos, if you watch videos or if you read textbooks or anything, you might see there are different information. This happens in metaphase and this happens in prophase. And so this is why they just made a phase called prometaphase. So it's kind of between both of them. Now at prometaphase or late prophase, now the chromatids, we said initially they were invisible, now they become distinguishable under a light microscope, so you're able to see the chromatids, nucleoli, and nuclear membrane disintegrate. Now this is very important because when you're dividing a cell, you really need the chromosomes to be present within the entire cell so you're able to divide it. If it's inside the nucleus, and it's very difficult to divide them, how are you going to divide them when there's a nuclear membrane present? You can't have a nuclear membrane. It would make the cell division very hard. And now the chromosomes, because we've removed the nuclear membrane, they're released into the cytoplasm. It makes sense because there is no nuclear membrane, so the chromosomes would go all over the cytoplasm. Now the spindle microtubules invade the central region of the cell. This also goes back to the point, I'm going to explain it more in metaphase, so they start going more towards the center. Now the chromosomes move from the center towards the equatorial plate or metaphase plate. Okay, I'll explain it in metaphase. Before I go into metaphase, did everyone understand prophase? Is there any question regarding anything I've said in prophase? No questions. Okay. Can you repeat prometaphase briefly? Sure. So basically, prometaphase is this phase between prophase and metaphase. It's also known as late prophase because after prophase comes metaphase. So this period of time where some textbooks would put this under metaphase and some would put it under late prophase. So there is another phase that's called prometaphase. It's not present in every textbook and every video, but you just should know and know this happens kind of between both of them. Late prophase at the beginning of metaphase. So this phase entirely is known as prometaphase. Now, initially in prophase, we said the chromatids were invisible, if you remember here. Now at the late prophase or prometaphase, now we're able to see them, distinguishable under light microscope. So we're able to see them. The nucleoli and nuclear membrane disintegrate and disappear. And this is important, as I said, because when you want to divide a cell, having a nuclear membrane would kind of limit your access to the chromosomes. We need the chromosomes to be in the cytoplasm so we're able to divide them equally into two different cells. Imagine if there's a nuclear membrane and the chromosomes are inside it, dividing the cell would be so hard. So what happens in prometaphase is this nuclear membrane disintegrates because we no longer have it. So now the chromosomes will go all over the place, all over the cytoplasm. So now it's easier for the cells to divide. Okay, sure. Sorry, I will speak slower. Okay, sorry. Okay. So we've disintegrated this nuclear membrane because it's no longer there. Okay, let's just delete it so it makes sense. We've disintegrated it. When you remove the nuclear membrane, all the chromosomes and the genetic material, they would go all over the cytoplasm. When they're all over the cytoplasm, now it's easier for the cells to divide because a nuclear membrane would kind of limit the access. We don't want to limit the access. We want to be able to divide the cell. When they're all over the cytoplasm, it's now easier for the cells to organize them and divide them. So this is why the nuclear membrane would disintegrate. And this happens in late prophase or prometaphase. Now, the last 2 points, I will explain them in metaphase because it's easier and it makes more sense to explain them there. Does that make sense so far? Did you understand the process so far? We disintegrated the nuclear membrane and now the chromosomes are in the cytoplasm. This is where we've reached, and the rest I'll explain it in the next slide. Understood. Okay. Okay, now we're gonna move to metaphase. I just want to make sure, Ahmed, did you understand what I said so far? If you have any questions, I'm so willing to repeat it. It's fine. Yes, okay, perfect. Now we're gonna move to metaphase. So there's chromosome alignment. We said so far we have 46 double-structured chromosomes. We said we have these chromosomes because we've replicated them in the S phase. Remember, it's replicated. We have 92 chromatids and 46 chromosomes. This is where we've reached. Now, these chromosomes, all of them align along what is known as equatorial plane, or also known as metaphase plane. Metaphase plate. Now, the metaphase plate is this imaginary plate that is present in the center of the cell. You see this dashed line in the center? This is what we call the metaphase plate, also known as the equatorial plane. Now, all these chromosomes would go and move towards the center. Okay, so they're all aligned in the center. This is what happens in metaphase. And they start attaching to the spindle microtubules. Remember, we said these spindle microtubules that were arising from the centrioles. Now, imagine we have centrioles here, and these mitotic spindles are coming out of them. Now they start attaching to the center of these chromosomes. To be more specific, they start attaching to the centromere, or the protein that we called earlier, the kinetic core. Remember, we talked about it, it's in the center of the chromosome. So this is where the mitotic spindles, which are these structures arising from the centrioles, they would go all the way and attach to the centromere or the kinetic core. And this all facilitates in the process of cell division. It makes sense because we want to attach so we're able to separate. Now, visibility of chromosomes, the double structure of the chromosome is clearly visible, just like the late prophase or prometaphase. In metaphase, the centromere is also a double structure. What this means, the centromere is attached to 2 chromatids, not attached to 1 chromatid, or like it's attaching 2 chromatids together. Does that make sense so far? And if you look here, we've already mentioned, the chromosomes are still like double in number. We have 92 chromatids and 46 chromosomes. So that's why we call it a double structure, because we have 2 chromatids rather than 1 attached to the centromere. Do you understand this so far? Okay, yes, perfect. Now that the chromosomes are aligned in the center in the metaphase plate or the equatorial plane, and they're attached to the mitotic spindles, what happens is these mitotic spindles start pulling the chromatids in like opposite directions. Sorry, someone asked, so a chromosome can have either 1 or 2 chromatids? Yes, exactly. A chromosome can have 1 chromatid and 1 centromere, and that's a chromosome. Or it can have 2 chromatids and 1 centromere, and that's again a chromosome. However, it has double the number of genetic material. The chromosome can have 1 or 2 chromatids, exactly. Okay, thank you. Perfect. You're welcome. Anyway, so yeah, chromosomes divide lengthwise. Lengthwise meaning يعني خلاص, they were aligned in this way, and lengthwise they've divided this way. If you look at the picture, it would make sense. Now, chromatids move towards the opposite pole of spindles. Spindle microtubules are responsible for moving chromatids to the poles, which makes sense because we said, no, they're going to pull them apart. Because also they were attached to the centromere or the kinetic core. So when they start pulling them in opposite directions, they will separate. A cleavage furrow begins to develop at this stage, but it's still not entirely developed. A cleavage furrow is this one here, where basically the cells begin to divide, particularly the cytoplasm of the cells. But it's not entirely developed yet, just beginning to develop. By the end, chromosomes are grouped at each pole of the cell, with both groups being diploid. exactly what we mentioned before. Now, why did we say both groups being diploid? Because we mentioned we had 46 chromosomes, but we had earlier, if you remember, 92 chromatids, because we've replicated the number of the genetic material in the S phase. Now, when we're dividing them, we would still have 46 chromosomes because each one of them does have a centromere. However, the genetic material would be halved. So instead of having 92 chromatids in each chromosome, now we'd have 46 in each cell. And this will be more obvious to us when we go to the telophase. I'll explain them together and then I'll ask if anyone has any questions about them. Now, the cleavage furrow is now entirely developed in the telophase, now, or process of cytokinesis. Cytokinesis is basically when you're dividing the cytoplasm into two different cells. And now the genetic material has divided, so we still have 46 chromosomes in each cell. Now we have 46 chromosomes and 46 chromosomes. Not only 46 chromosomes, but if you look here, the chromosome is no longer having two chromatids, it has one chromatid. So we have 46 chromatids as well. So we've halved the genetic material, but the number of chromosomes remains the same. We have 46 chromosomes and 46 chromatids. Now, because we've already divided the cells and we want to end up with having two cells, now the nucleus reappears, the nuclear membrane reappears, nucleolus reappears, and new centrioles are formed in each cell, يعني, and the division of cytoplasm occurs, and خلاص. Now we're almost done with mitosis. Does everyone understand everything we've talked about in anaphase and telophase? I'm willing to repeat anything if anyone didn't understand. All clear. Okay. Okay, perfect. Now I'm going to go over everything briefly so that it makes sense. So I'll be as quiet as possible and sorry, as slow as possible, so you can grasp everything I say and خلاص, you will see it all in one picture. So normally we said we have a cell and we said the genetic material in the cell is very loose and it's like just all over the place in the nucleus. And in order for us to be able to divide the cell, we need to condense the genetic material. So what happens is the cell enters mitosis. In prophase, the genetic material begins to become more condensed. And this is important so that we're able to organize the cell cycle and خلاص, يعني, decrease the chances of error. After prophase, we have the prometaphase or the late prophase. And we have the chromosomes. We have 46 chromosomes and 92 chromatids. And why do we have 92 chromatids? Because we've already replicated the genetic material in S phase, which is in the interphase, before getting into mitosis. So we have 92 chromatids, but 46 chromosomes. Now we go to metaphase. In the metaphase, you can see that the chromosomes are all aligning in the center and what is known as the metaphase plate or the equatorial plane. And the chromosomes are attaching to these mitotic spindles. Now in anaphase, these mitotic spindles would kind of pull each chromatid in the opposite pole. And now we have a chromatid going to this side and a chromatid going to that side. So now the genetic material will be halved in number. Instead of having 92 chromatids, we would have 46 chromatids, but we still have 46 chromosomes because we said what determines the number of chromosomes is the number of centrioles, and each one of these chromatids does have a centriole. Now we're going to go to telophase. In telophase, we form what is known as cleavage furrow, which divides the cytoplasm of the cells, forming two cells. And this process of dividing the cytoplasm is what we call cytokinesis. The end result would be two daughter cells that are exactly identical to the mother cell and 46 chromosomes in each one of them. Does that make sense so far? Did you understand the steps of mitosis? Can you explain how it relates to the G1, G2, etc.? Sure. I'll go back to the slide. Okay. So here, this one is good. So this G1 S phase and G2 phase, this entire thing is what we call the interphase. Now the interphase is the process, I'm just going to write this, is the process where the cells prepare to go to the actual cell division, which is either mitosis or meiosis. Now what happens is in G1, the cells start to acquire their function, they start specializing, because initially our cells are stem cells, and then they start to kind of decide, okay, I'm going to be an epithelial cells, I'm going to be an intestinal cell. So this process of like deciding what they're going to be or like specializing happens in G1. And then in the S phase, this is when the DNA is going to be replicated. So instead of having 46 genetic material or 46 chromatids, we'd have double the number, which is 92. And then the G2 phase is mainly responsible for like checking if there are any errors before going into the cell division. So because we've just replicated our DNA, it's important to have another step that checks, did we replicate it correctly? Were there any errors to prevent any genetic diseases? So this is where G2 comes into play. It's mainly to double check that there were no errors in the replication, and then خلاص, the cell would undergo mitosis or meiosis depending on the cell phase. Now, another question, in anaphase, is it 46 chromosomes and 92 chromatids? Let's go to anaphase, 1 second. So in anaphase, it is the process of where you're dividing your 92 chromosomes to 46. So this is not a question that would come because honestly, it starts as 92 and then it divides into 46, but yet it's not entirely divided. Yani it's in the process of dividing the 92 into 46, if that makes sense. So they wouldn't ask you how many because it's just in the process of splitting the 92 into 46, of course. But the one for sure, it is 46 chromatids is the telophase because خلاص, we've divided them and we're now undergoing the cytokinesis. Okay, is that clear so far before we go into the question? Does the centromere divides? The center, I wouldn't call it divides. It's just, it's not entirely dividing. It's just when you have two chromosomes attached on one centromere, when they undergo like division into two chromatids, each one of them would have a centromere. So you can understand it in the concept of dividing, but it's not entirely dividing. Like dividing is not the word for it. It's just they would split from two chromosomes to individual chromatids, but they would still have a centromere. It's not entirely dividing. But yes, if you want to understand that each chromatid has a centromere, then yes, it does. After having one, they go into two, but it's not really division. Does that clear your question? Okay, another question is when they're joined together, are they considered one chromosome or two chromosomes with two chromatids? No, they're considered one chromosome. I mentioned earlier with the sites, the number of chromosomes is the number of centromeres. So even if you have two chromatids joined by one centromere, it is a one chromosome. If you have a million chromatids and it's just one centromere, it is still a one chromosome. What decides the number of chromosomes is the number of centromeres. So you don't really care about how many chromatids we have in a chromosome. What we really care about is how many centromeres. This tells you how many chromosomes you have. So yes, you can have one chromosome with two chromatids or one chromosome with one chromatid. Of course. Does anyone have any other questions? Okay, seems that there are no questions, so I'm going to ask you. I'll give you a minute to solve it, and then please send your answers in direct message, not to everyone, so you don't cheat from each other. Ignore the question mark here. Oh, I'm sorry. Sorry. This is a question about meiosis. I just put it in the wrong place. Also, I'll ask you when we go to meiosis. Ignore these questions. I'm sorry. These are about meiosis. I put them in the wrong place. Okay. I'll ask you about them after meiosis. We're going to go back to these questions and explain them, inshallah. Now, since everyone understood mitosis, now we're going to go to a little bit more complicated, but it's not hard if you understood mitosis, which is the process of meiosis. Now, meiosis, as I mentioned earlier, it occurs in sex cells or gametes. It consists of two cell divisions. We have meiosis I and meiosis II, whereas mitosis was just one division. It is also known as reduction division. Now, the reason it is known as reduction division because we start with 46 chromosomes, but we end up with 23 chromosomes. So we have reduced the number of chromosomes from 46 to 23. And we're going to explain the details and how this happened in the upcoming slides, inshallah. Now, there are three major things that are happening in meiosis. Just like mitosis, DNA replication happens prior to going through meiosis itself. So before meiosis begins, DNA is replicated, and this happens during the S phase of the interphase, which results in tetraploid DNA content, which means we have 92 chromatids so far, just like mitosis. Now the chromosome number remains diploid. So we still have 46 chromosomes, but we have 92 chromatids. Why? Because each two chromatids will form one chromosome. Does that make sense so far? Because this point, I believe, can be confusing to some of you, and this point is the one I got most questions about. Do you understand the concept of having 92 chromatids and 46 chromosomes? Yes, okay. Okay, perfect. Now, after the DNA replication, we're going to go through meiosis I. Now, DNA amount is half to diploid level because we started with tetraploid, which is 92. Now we're going to have it to 46. I'll explain how in the upcoming slide. I'm just giving you an overview. Now, the chromosome number is reduced from diploid to haploid. Again, I'm going to explain in details how, but the chromosomes are still double structured. I'll draw the whole process. And then in meiosis II, everything will be haploid. So the number of chromosomes is now 23, and the number of chromatids would be 23. So this is the end result. We'll explain how this happens in more details. So the first phase of meiosis is known as prophase. Now this prophase is subdivided into 5 phases, which are leptotene, zygotene, pachytene, diplotene, diakinesis. To be honest, we did not take this when we were in first year. So I'm not sure how deep they're going to ask you. So I just added everything that was in your slides, and I'm going to explain that. I'm not going to go deeper than that because as first years, we've never been exposed to these concepts. I just studied them for you guys and to be able to explain them. But I personally, even though I'm a fourth year, I've never heard of them before. So I'm not sure how important they are for your exam, but other than foundation exam, you'll probably never hear of these 5 phases ever. But I'm just going to explain them for the sake of your lecture. Okay, so now we're going to go to the leptotene phase. Now, the leptotene phase basically is very similar to the initial phase of prophase, that the chromosomes start thin and then become shortened and thickened. Now we said, we need the chromosomes to be thick and condensed to be able to divide the cells. So the same concept applies here. In leptotene phase, the chromosomes begin to shorten and condense, and this is important. They appear as individual threads, which is kind of showing here in this image. Just imagine that they look like that, just threads. They're attached to the nuclear envelope at one end. So if you see here, this thread is attached to the nuclear envelope at this one end, but the other end, it's not attached to it. So this is just explaining the appearance. It shows a beaded appearance. Each bead is called a chromomere. Now, if you look at this picture, you don't need to know the picture. I added it for you to understand. You see these lines here? These lines that are present along the chromosome is what's called what's known as a chromomere. Now, these chromomeres are present at the leptotene phase. The way to study these phases, always remember, this happens in which phase? Which can be challenging in the beginning, but if you just organize it in your head and make it, it will make sense to you. So initially, it starts as being condensed, and whilst, the way I imagine it, and while it's condensing, we're gonna kind of, when you're shrinking something, you would start having lines. So this is what's causing the beaded appearance. And now the asters and spindle fibers form. If you remember, the spindle fibers are the ones that are originating from the centriole. So these are the mitotic spindles or the spindle fibers. They can be called either. And the asters are the ones that are attaching the centrioles to the opposite pole. Now, the nucleolus and the nuclear envelope so far are still present. We did not disintegrate them yet. So this is mainly what's happening in the leptotene phase. And don't forget that it's a subdivision of prophase 1. Okay. Now, after the leptotene phase, we're going to go to the next phase, which is called the zygotene. Now, pairing occurs in this stage. Basically, what this means, let me just draw it. I'll take a different color. Now, what happens in meiosis is that you're going to have one pair of chromosome coming from the mother. Okay, let's say this, the red represents the maternal chromosome. And the blue represents the paternal, which means coming from the mother, from the father, sorry, the paternal chromosomes. So this one is coming from the mother, this one is coming from the father. Anything that's coming from the mother can be called maternal. Anything that's coming from the father can be called paternal. Now these two chromosomes, because we know in meiosis, we're forming, it's a division of a sex cell. So these will join together. Now pairing means like pairing of these two chromosomes together. Each pair is called bivalent. Now, why is this important? Why do we need to know this terminology? Because bivalent refers to two chromosomes together. So bi stands for two chromosomes, but we have four chromatids. So this process of like forming them together is called pairing. This concept is kind of not easy to understand, but I just want you to just imagine with me that this is a sex chromosome. So one chromosome will be coming from the mother and one chromosome will be coming from the father. And this makes sense because, sex cells arise after fertilization. So we have one from the mother and one from the father. And when they're joined together, this process is called pairing. Collectively together, we call them bivalent. Now, bi refers to the number of chromosomes, not chromatids. We have two chromosomes, but we have four chromatids. Now, one chromosome comes from the father and the other from the mother. This process is called synapsis or conjugation, which is basically the pairing process, can be also called synapsis or conjugation. They all kind of have similar meaning. You're synapsing two things together, conjugating, pairing. So they all refer kind of to the same point, which is they're both coming closer together and forming like pairs. Pairing is exact and occurs point to point, starting at the attachment to the nuclear membrane and moving downward. So this what it's trying to tell you basically is that remember the karyotyping picture that I showed you. If you see these chromosomes, you would notice that each pair has very similar chromosomes. This chromosome is very similar to this one, this chromosome is very similar to this one, and each one of them is inherited either from one from the mother and one from the father. So what it's trying to tell you is that the pairing point to point, meaning the chromosomes that are very similar to each other are the ones that pair, and it's like the pairing occurs at the now the centromeres do not pair, as I drew here. We have a centromere in the center and a centromere in the center. Now the pairing occurs throughout the chromosome. Sorry, let's delete this line. The pairing occurs throughout the chromosome, but not in the centromere. So it would pair here, 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 all over, but it wouldn't pair in the centromere. This will become more significant later on. I'll explain it when we get to that point. Now, each homologous pair is identical in length, as I showed you in the karyotyping, centromere location, and banding pattern. Banding pattern is basically whether they're shaped this way or this way or this way, whatever shape they are, the pairs would be identical. And you would see them having similar appearance this way. That's what it's trying to tell you. Since each chromosome is double structured with two chromatids, the homologous pair has a total of 4 chromatids, which is literally what I drew here. The chromosomes are double structured, meaning they have 2 chromatids. This is one chromatid, and this is the other one. So this pair, or this what we call as bivalent, I'll just draw bivalent, okay? So they have 4 chromatids and 2 chromosomes. So far, nothing really happened. We're just saying, it's a sex cell that arises after fertilization. So we're gonna have 1 chromosome from the mother, 1 chromosome from the father. They're getting close together. This process where each pair gets close together, each similar pair, is called conjugation, synapsis, pairing. And the outcome would be a structure called bivalent. The bivalent has 4 chromatids making up 2 chromosomes. Does that make sense so far? That's all I want you to know so far. We have 2 chromosomes. In each one of them, we have 2 chromatids, so a total of 4 chromatids. And this structure together, when they're pairing and like coming close to each other, is what we call a bivalent. Does that make sense so far? Yes, yes. Okay, perfect. Okay. Now that was the case for all the autosomal chromosomes, meaning the 22 pairs of chromosomes that I showed you in the karyotyping. Now, what happens to the X and Y chromosomes? Because now we know for a fact that the X chromosome is different than the Y chromosome. So how are they going to pair when we said the pairing occurs point to point, and they're very similar structures? Now, when it comes to the X and Y chromosomes, we have different segments in each one of them. So the similar segments which can pair is what we call the pairing segment. So this is the X chromosome. Okay, so let's look at this picture. Just have the red. Okay, this is the X chromosome and this is the Y chromosome. The homologous segment or similar segments is what we call the pairing segment. So they're able to pair together. However, the remaining parts are called the differential segments. These parts don't pair together. So the X and Y chromosomes are found in a special area of the nucleus called the sex vesicle. This is just nothing really significant except it keeps them like close to each other. Now the homologous chromosomes, this applies to both autosomal and X and Y chromosomes. They're held together by a structure called synaptonymal complex. Now this synaptonymal complex tells you, they're not only close together, but there's also something that's holding them and keeping them close together. And this structure is what we call the synaptonymal complex. This will become more significant in the upcoming slides. Just remember its name. So what's keeping these chromosomes close together is a structure called synaptonymal complex. Does that make sense so far? And do you understand everything in the zygote team? Yes, okay, perfect. Now we're gonna move to the Pachytene stage. Now, again, as I said before, when you study these lectures or these slides specifically, try to remember which happens when. This is how the questions would usually be asked. So an important thing that happens in the Pachytene stage, which is, remember, it's a subdivision of prophase 1, is what's known as the crossing over. Now, chromosomes continue to shorten and thicken, and pairing is complete, which is something we've said before. Now, chromatids become visible and attach at the centromere. Again, it makes sense. Each chromosome pair forms a tetrad, 4 chromatids, which again, remember the chromosomes that I just drew earlier, the one that's coming from the mother and the other that's coming from the father. So we can either call them a bivalent, and when we say bivalent, we mean there are 2 chromosomes that are made of 4 chromatids. But in the context of chromatids, we can call it a tetrad. Tetra means 4. So we have 4 chromatids, but they're all referring to the same structure. We can either call it bivalent, meaning we have 2 chromosomes each having 4 chromatids, or we can call it tetrad, which are 4 chromatids. But it's the same structure, just different names for the same structure. Now, crossing over occurs as 2 chromatids form from each pair partially coil around each other. Now, if you look at this picture here, it would make it, make way more sense. Now, these are, let's take these chromosomes and let's take them here. So these are the homologous chromosomes aligned. I need the one coming from the mother and one coming from the father. Now, from each chromosome, one chromatid, so this chromatid here and this chromatid here, would basically start to undergo a process that is called crossing over. And by crossing over, they would exchange some genetic material. So if you see, parts of the blue would go to the red, and parts of the red would go to the blue. Now, this is important because this like process of crossing over, it only happens in meiosis. It does not happen in mitosis. And this is the reason why the daughter cells are not identical to the parent cell. Yani the parent cell and mitosis would produce 2 identical daughter cells, but in meiosis, it would have 4 different daughter cells, and this is because of the process of crossing over. Someone is asking me, are the yellow dots centromeres? Because if they are, wouldn't there be 4 chromosomes and 4 chromatids? Okay, now they're trying to represent centromere, but this is not an accurate representation. This is just a picture to make you understand. Don't take, just imagine if it's 1 centromere, because these are 2 chromosomes. Okay, this is not an accurate picture. And I took this from the internet just to make the process of crossing over clearer, but I just want you to imagine. I'll edit this when I send you the slides. And no, it's just 1 centromere. I didn't focus on the centromere part because I was mostly focusing on the crossing over. But you're right, it shouldn't be 2 centromeres, it should be 1. Because this is 1 chromosome and this is another 1 chromosome, 1 coming from the father and 1 coming from the mother. Does that clear your point? Of course. Okay, so we said crossing over happens at the Pachytene stage, which is part of prophase 1. Because a question can come to you asking, someone is asking if I'm going to post the recording. Yes, it will be posted on YouTube, inshallah. Anyway, so yeah, crossing over happens and there's exchange of genetic material from each chromatid. During crossing over, DNA exchange happens through breaking and rejoining, which makes total sense. Now we have what is known as the synaptonymal complex. It facilitates this process. And remember the synaptonymal complex that I said here? It basically holds the structure close together. So crossing over would not happen if the structures were not close together. So a question can also come and ask you, which structure facilitates the process of crossing over, then it would be the synaptonymal complex because it's keeping them close together, allowing crossover to take place. Another important thing to remember in a crossing over can happen in Pachytene stage or prophase 1. Yani the answer can be either Pachytene or prophase 1. Why? Because Pachytene is a subdivision of prophase 1. And it's actually more high yield to ask إنه it happens in prophase 1 than saying Pachytene stage. But remember them both for the sake of your lecture. But generally speaking, يعني if it's just other than foundation, any other exam, they would probably ask you, and a crossing over happens in prophase 1. Okay. Now, see these regions where DNA has been exchanged after the crossing over? They're marked by proteins that are called recombination modules. So this is just a name protein that is present near the areas of crossing over. So there isn't much really to understand except another protein that are called recombination modules near the areas of crossing over or the regions of DNA exchange. Do you understand everything that's happening in Pachytene stage? And I'm more than happy to repeat if someone didn't explain anything. Sorry, didn't understand anything. What does recombination modules do? They don't really contribute significantly, but it's just, and what you really need to know for the sake of your lecture, they're just present near the areas of crossing over. And whenever there is crossing over, you would see these proteins, the recombination modules. Perfect. Does anyone have any other questions? Okay. Since no one has questions, we're going to move to the diplotin stage. Now, in the diplotin stage, the homologous chromosomes begin to separate except the regions where crossing over has taken place. Yes, another important thing to note is that the regions where crossing over takes place, which are these regions, are known as chiasmata as plural and chiasma as a singular. So this region is called chiasma. These two regions are called chiasmata. So you just know the name. This is the area of crossing over. So in which area does over happen? It's just chiasmata or chiasma. And yes, they begin to separate because that's just the process of, they were attached together, now they're separating. Typically, each pair of chromosomes. Yes, so at least one chiasma happening, at least one crossover reaction happens in a pair of chromosomes, but it can go up to 5 or 10 crossing over reactions. So you can have up to 5 or 10 chiasmata in one pair of homologous chromosomes. Does that make sense so far? There isn't really much to explain here. It's just mostly terminologies. The separation will be discussed more later, but do you understand everything here so far? And just they're called chiasmata, and in every homologous pair, at least one chiasma must be present. One crossing over reaction must happen in every homologous pair of chromosomes. Yes, okay, perfect. Now we're going to move to the last stage of prophase, which is called diakinesis. The remaining chiasmata are resolved, chromosomes continue to shorten. Now, everything here that's happening, it's just preparing for the upcoming stage, which is the metaphase. Now, the chiasmata are resolved basically in khals. We've already had the crossing over, and then they continue to shorten because it continues to condense. Centromeres move further apart, and this is allowing for the chromosomes to divide, which will happen later on. Chromosomes are now arranged as bivalent, which we explained what bivalent is. It's basically a pair of 2 chromosomes and 4 chromatids. They position themselves near the nuclear envelope, as shown in the picture, and extreme segments of the chromosomes remain attached. So even though the chiasmata began to move, because we said it's resolving, they're still attached a little bit together, as shown here. Does that make sense? And honestly, there isn't much to explain. Just telling you continuation of what was happening. It's just like a storytelling. I don't think there's anything significant, honestly, in diakinesis. Now, to continue talking about meiosis I, we finished the prophase with all its subtypes. Then we have the prometaphase, which is exactly the same as in mitosis. So there's really no need to explain it because we've already understood that in mitosis. Then the metaphase one is the same in mitosis except for, okay, now let's understand what's the difference. Now this is very important. Here I want you to focus. In metaphase, normally, if you remember, we said, let's talk about mitosis. Everything I'm drawing here in red, it's mitosis, okay? نحن we said initially in mitosis, we started with 46 chromosomes and it had one chromatid. Then we replicated the genetic material. So we had 92 chromatids, but still we had 46 chromosomes. صح? So I'm just going to draw it here. And then in the metaphase of mitosis, we had them aligned in the metaphase plate, which was in the center. صح? If you remember. This is how it looked like. And then we had the microtubule pulling them apart. So we had the end result would be something like this. And then we had 46 chromosomes here and 46 chromosomes here. Each one of them, let's imagine these are two cells, each one of them having 46 chromatids. Do you remember this was what's happening in mitosis? Now in meiosis I, to be particular, I'm gonna draw it and actually let's make it blue. What happens is, remember we said the chromosomes are now aligned in pairs or what is known as bivalent. So this also applies to the metaphase. In metaphase, we would have pairs of chromosomes aligned. So we'd have this paternal chromosome and one maternal chromosome, which we call basically the bivalent. And it continues like that. Okay. So they're all aligned in the metaphase plate. And what happens when they divide, so here we have, here is meiosis. So here we have these each bivalent pairs of chromosomes present in the center. And they're all 23 pairs of chromosomes, meaning there are a total of 46 chromosomes, but we have 23 pairs of them. What happens next is that the cell would start to divide. So let's say we have, after meiosis I, we're going to end up having two cells. But the division this time will not be in like single chromosome with a single chromatid. لا, this time it's going to be a chromosome going here. Sorry, and a chromosome going here. And a chromosome going here, and a chromosome going here. So that's the major difference. Here it's saying, 23 pairs of chromosomes align along the equator. The equator is basically the metaphase plate or the equatorial plane. So if you look at this picture, come on, it's showing it, and you're going to have one maternal chromosome and one paternal chromosome. These bivalent structure or two chromosomes are aligned in the center. It's not one chromosome, it's two. The bivalent or this structure here. And now each homologous pair is arranged parallel to the equatorial plate with one member in either side. So we said one member is on the right side, which is the maternal side, and the other one is the paternal side. Does that make sense? This is the major difference in metaphase. The two pairs of chromosomes are present in the equatorial plane or the metaphase plate rather than just one chromosome present, which is seen in mitosis. Does that make sense? Do you understand this concept? And we have two chromosomes or a pair of chromosomes at the metaphase plate. Yes. Okay, perfect. Please stop me if you have any questions. I'm more than happy to answer, but I'm gonna proceed because it seems like you guys understood, alhamdulillah. Okay, so now we're gonna move to the anaphase one. Now, anaphase in meiosis is similar to that in mitosis with some key differences, which is very similar to the previous difference. Instead of separating 46 double-structured chromosome, the homologous pairs separates. We said before, here we're separating the pair of 46 chromosomes in mitosis. So one would go here, one pair would, one chromatid would go here, and one chromatid would go here. However, in anaphase in meiosis I, you're going to see, each chromosome would go to the opposite side. Basically, we're dividing the homologous pairs with one member of each pair moving to the opposite pole, which is what I just illustrated, and it's also shown here. You see, a whole chromosome is going to the opposite side. It's not just a chromatid. The rest of the events are similar in mitosis. Now, the centromeres do not split. Instead, the adherent ends of the chromatids separate. Okay. Now, the telophase one is the same as mitosis, which basically, there's cytokinesis that's going to happen, and then we're going to have two cells as an end result of meiosis I. Now, as a result of the first meiotic division, two new daughter cells are formed, which we just illustrated here. Now, the cells would have diploid amount of DNA, which means we have two chromatids present in the chromosome. meaning 46 chromatids. However, we'd have 23 pairs of chromosomes, so we would still have a haploid number of chromosomes. So after meiosis I, we would have a haploid number of chromosomes because there are 23 chromosomes. However, we have 46 chromatids. Does that make sense? Because similar to the concept that I've been explaining earlier, because it's still meiosis I. We didn't go through meiosis II. In meiosis I, we have a haploid number of chromosomes, meaning 23 chromosomes. But inside the chromosomes, we have 2 chromatids, meaning we'd have 46 chromatids. Does that make sense so far? Yes, okay, perfect. Now we're going to start by talking about meiosis II. If anyone has any question about meiosis I, this is the perfect time to ask, so I can go back and explain it before getting into meiosis II. So I'm just going to emphasize, if you have any questions, please feel free to ask. Okay. Okay, so I'm going to start with meiosis II, which occurs shortly after the first division. Now we divided the cells in meiosis I. We're going to go into meiosis II. Now, no DNA synthesis takes place before the division. If you remember, in meiosis I, we went through the S phase. We've replicated the DNA, then we went through meiosis I. So there was a DNA replication here. We don't replicate the DNA after meiosis I. Plus, we were left with 23 chromosomes and 46 chromatids. This is where we're going to go on from. There is no DNA replication happening. Now, the 2 daughter cells contain 23 double-structured chromosomes, and the DNA quantity is diploid, but the chromosome number is haploid, which is literally what I just said here. We have 23 chromosomes, but the chromatids, or the DNA quantity, is 46. Now, all phases of division are similar to mitosis with some differences. The separating chromatids are genetically different. And can someone tell me what was the process that happened in meiosis I that made the chromosomes genetically different and not identical to the parent cell? Crossing over, exactly. It's the crossing over reaction that made the chromatids genetically different than the parent cell. Exactly, bravo. Now, the 23 double-structured chromosomes align along the equatorial plate instead of 46, because if you remember, in mitosis, we had 46 chromosomes in the center. Here, we would have 23 rather than 46. In meiosis I, we had the bivalent structure, so 2 chromosomes. Here we have 1 chromosome each time, but they're only 23. In mitosis, they were 46, because mitosis, it was diploid cells. Here we were dealing with a haploid cell. The daughter cells have 23 chromosomes, haploid. The amount of DNA is also half. That's after undergoing meiosis II, which will explain how this happens. Okay. Now, the purpose of 2 meiotic divisions, and with exchange genetic material and create new chromosomes with are genetically similar, okay, which we said, crossing over basically, to provide each germ cells with both haploid number of chromosomes and haploid number of DNA. And this is important. We need the germ cells to have a haploid number of chromosomes and haploid number of DNA. Both need to be haploid, because the whole purpose of a germ cell is basically to undergo fertilization and join another germ cell. So a sperm would join an ovum. So we need them both to have 23 chromosomes. So when they form a baby, it has 46 chromosomes, which is basically a somatic cell. Does that make sense? Why we need to have them haploid number? Because they are sex cells. They have to be haploid, because eventually they're going to fuse and like fertilize together. And we can't have excess number of chromosomes. Does that make sense? Why we need the germ cells to be 23 chromosomes? The whole purpose of having 2 meiotic divisions. Like when you understand why it happens, it just makes way more sense to study it. Yes, okay, perfect. Now there's something that is known as abnormal meiotic divisions. Mitotic and meiotic divisions. Honestly, these abnormalities can happen in both of them. There is what is known as non-disjunction. Now, non-disjunction is basically a problem in separating chromosomes. Because if you look back at the slide, we just kept saying, the chromosomes would divide, the microtubules or its spindles would like just separate them. So what would happen here, so look at this picture, this is a proper disjunction. Disjunction meaning they're not junctioned together, you're separating them. That's what disjunction means. Non-disjunction is that when you're not unable to separate them, so instead of having this pair coming here, where it's supposed to be, it would just go to the opposite side. And this is basically what results in trisomies. So if you look at the perfect case scenario, the person should have, for example, let's say 2 pairs, for example, these are theoretical numbers, 2 pairs in each cell. If there is non-disjunction, this process basically just happened. I'm just going to erase what I drew so it makes more sense. Okay. If there is non-disjunction that happened, what would happen is 3 would go here and 1 would go here. And this is basically what results in syndromes, abnormalities such as Down syndrome, Edwards syndrome, and so on. So the daughter cells would end up having unequal numbers of chromosomes, and that's obviously not good. Now, the lagging of chromatids does not necessarily result in this abnormality. So lagging of chromatids is basically the way you can see it is lagging of chromatids can lead to non-disjunction. It's not an abnormality on its own. Lagging chromatids does not always result in abnormalities, but basically it can increase the risk of having non-disjunction, which causes an abnormality. Lagging of chromatids is basically the chromatids would lag while you're separating them, causing a problem. But this doesn't always end up having a problem. The body can sometimes resolve this issue. But non-disjunction, it always leads to a problem. Do you understand that? Can you repeat? Sure. So if you see here, when we're dividing the chromosomes with the mitotic spindles, one should go here, one chromatid, and one should go here. Sometimes the chromatid, after they're being separated, it can lag, meaning basically when you literally lag, just the word lag. It's just when the mitotic spindles are pulling them, they're just lagging. But the body can sometimes resolve this issue, and every chromatid would end up going to its opposite pole normally. It's just lagging. What's really not normal is the non-disjunction, because non-disjunction is the chromatid to begin with didn't go to the opposite side, so it just went to the other side, which resulted in abnormal number of chromosomes in one of the cells. Now, what I'm trying to say here is that lagging of chromatids can contribute to non-disjunction, because if it really lags, then it wouldn't come to the opposite pole, so it's really not separated, contributing to non-disjunction. However, the body can potentially resolve. So lagging of chromatids does not always result in an abnormality, but a non-disjunction always results in an abnormality. So non-disjunction is kind of the final result. Does that make sense? Yes, okay, perfect. Does anyone have any questions so far? Is it all clear? Okay, since it's all clear, okay, I have one question. If the presence of one pair from the mom and one from the dad in meiosis meant that fertilization happened, why is the result of meiosis a germ cell without which will undergo fertilization? Okay. Okay, you're asking a very deep concept. I will explain it. This will make more sense when we go to oogenesis and spermatogenesis at the end of this lecture, but I'll try to briefly explain it to you now. So basically, she's asking if the presence one pair from the mom, which we explain here, this is the mom, and one pair from the dad, meant that fertilization happened. Of course, it happened because basically a mom and dad, I'll just explain it in simple terms. A mom and dad, okay, basically a fertilization process has occurred and now they're forming a baby. So the first cell in a baby is what we call a zygote, which basically when the two cells join together, when a sperm joins an ovum, we will have a zygote. I don't know if you know that, but let's just call it a zygote so you understand. Now this zygote will eventually grow, and become a human being. Okay, basically the cells will keep dividing and it will become a human. It wouldn't become any germ cell. But the thing is, what I was referring to, and why is the result of meiosis germ cell which will undergo fertilization? When we talk about meiosis in the context of oogenesis or spermatogenesis, which basically is meiosis, you will need to have the germ cell in particular. The zygote, okay, I'll explain it in a simpler term. The zygote, it came from one haploid cell and one haploid cell. They join together and they form a diploid cell, okay? Diploid cell. This is us. We started as zygotes. We started as diploid cell. One haploid cell came from the father, which is a sperm, and one haploid cell came from the mother, which is the ovum, and it formed a diploid cell, which is the zygote, which made us as humans. Now, what I'm referring to, and it has to be haploid, I'm referring to the sperm and the ovum itself. Now, if the ovum of the mother was not 23 chromosomes, it wouldn't make sense because it's making us, and we're humans who are made of 46 chromosomes in all the cells in our body. So the ovum of the mother has to be haploid and the sperm of the father has to be haploid too. So when they join and they form a human, it's 46 chromosomes. Do you understand this? If you don't, it'll be way more clear when we go to spermatogenesis and oogenesis later on in the lecture. And I will get back to your question in the process of their division. So one is asking, so non-disjunction, we don't be able to separate them, but lagging, it will be slow. It's not that it would be slow, it's just it would, the chromatid separation, it would be not as smooth, let's say, as normally would, which can increase the risk of having non-disjunction. And non-disjunction, yes, we won't be able to separate them. It's the end result. There is no non-disjunction, of course. Okay, for your question about the... Okay, 1 second. Yeah, about the presence pair from the mom and dad. Inshallah, we'll get back to it. And spermatogenesis and oogenesis, it'll make way more sense. But I understand what's confusing. Now we're going to go back to the questions. I hope you guys didn't already see the answers. But yeah, can you try and answer these? There's another question, inshallah, I'll answer it after we answer these questions. Okay, so I see A, D, A, and one B. Another D, B. Okay, I think A. I can see that it's confusing you a bit, so okay, the true answer is actually A. And the reason is, let's go back to Meiosis I. Oh, it's after. This is not the picture I was looking for. Let's go back. Okay, I have a better picture that will show it. Yeah, here. If you look at meiosis I, so we undergo interphase, then prophase 1, the prometaphase, everything, and then we have the telophase where we divide them. If you remember, we said we're going to start with 46 chromosomes in this cell, but we'd have 92 pairs of chromatids. Now the end results would be 23 chromosomes in this cell and 23 chromosomes for the cells. And when we say haploid, we're referring to the number of chromosomes. So after meiosis I, it is a haploid cell. We have 23 chromosomes. So let's go back to the questions. How many chromosomes are present after meiosis I in a diploid organism? Diploid organism is basically it's just there to confuse you. It would be 23 chromosomes because we've already divided them. We don't care. And we said we don't care about the number of chromatids when we say chromosomes. We care about the number of centromeres. So we'd end up having 23 chromosomes. The chromatids are really not relevant in whether it's haploid or diploid. So they're asking you, is it a chromosome? About the number of chromosomes. So you'd say 23. Diploid organism is just there to confuse you. Diploid organism is just, it's just like saying a human, basically. Aren't B and D same options? No, it's not. Because 23 chromosomes is different than 23 pairs of chromosomes. 23 pairs means 46 chromosomes. Oh, B and D. Oh, yeah, B and D are the same answer. Sorry, I thought you meant A and D. Yes, B and D are the same option. What about meiosis II? It is also 23. I'm a little confused. Yes, meiosis II is also 23 chromosomes, but the difference is we'd have, rather than 40, let's go back to the lecture, to the picture I drew in. We said so far, we would end up with 23 chromosomes, but we would have 46 chromatids. Remember, we said the number of chromosomes is decided by the number of chromatids. By the end of meiosis II, the number of chromosomes remains the same, but the number of chromatids is now also 23. So to make it clear, I'll just draw it for you guys. So we start here. I'm just going to start from metaphase because this is where I believe it matters. So this is meiosis I. We have metaphase. We said we'd have 2 chromosomes aligned, okay? And then we're going to divide them. When we divide them, here we started with 46 chromosomes. When we divide them, we're going to have 23 chromosomes, okay, in each cell. But if you notice, each chromosome has 2 chromatids, meaning we would have 92 chromatids so far. So here we started with 92 chromatids. And here we would have 46 chromatids, but 23 chromosomes. You need to know the difference. When we say the number of chromosomes, it's not the same as the number of chromatids. When they undergo meiosis II, what would happen basically is that these chromosomes would basically divide like that. So you'd have 23 chromosomes as well as 23 chromatids. So everything's now haploid. Do you understand this? So yes, Rasha, your answer is actually really good. She said 23 chromosomes with 46 chromatids. Yes, exactly. This is what happens after meiosis I. If a question said chromatids instead of chromosomes, answer would be D. Yes, it would be D. But they're less likely to ask you about chromatids. They mostly, in these questions, they would ask you about chromosomes rather than chromatids, if that makes sense. Okay? Do you understand the concept? And the person who asked, and aren't B and D the same option? Yes, they are. 23 pairs basically means 46 chromosomes. Do you all understand this question? If you don't, it's fine. I'm more than happy to repeat. I just want to make sure that you all understand it. Because questions are what can clear the confusion. And I added these questions on purpose because I knew it would confuse some of you. So it's good that you are engaging in them. So you know where you're not understanding exactly. Any questions test your knowledge. Do you understand? Okay, perfect. Does anyone have any other question or should I move forward? Seems like all clear. Okay, so I'm going to move to the next question. What are chiasmata? I've mentioned it earlier. I understand this might be a bit confusing because you're still new to the terminology, but yeah, someone said B. Okay, B, 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 B. A lot of B's. Exactly, bravo. The answer is B. Chiasmata are the points of crossing over between homologous chromosomes. Alhamdulillah, you guys all answered it well. Alhamdulillah. Okay, perfect. Now, does anyone have any questions so far about anything about mitosis, meiosis, 1, 2, any of the phases, anything that's confusing them? And now is the perfect time to ask because I'm going to move to the other part of the lecture. It's completely other part. So if you have any questions about anything I've explained, I'm more than happy to repeat it now. A girl asking me what's the difference between a bivalent pair and homologous pair. Okay, this is a very good question. Let's go back to the karyotyping side. So you see these pairs and the karyotyping. This is what we call homologous pair. Homologous pair is basically one chromosome coming from the mother and one chromosome coming from the father. And if you see, it only has like one chromatid. So that's a homologous pair. Okay, it's one chromatid and one chromatid. And like, this is the homologous pair. So you see 1, 2, 3, 4, and we have like the rest of them. So one chromatid making one chromosome from the father and one chromatid making one chromosome from the mother. This is what we call a homologous pair. And they're the ones that undergo crossing over and everything. Now, when we say bivalent pair, we're more specifically saying, I know there's, wait, let me just, because I've used these colors before, I'm gonna use the same colors. It's like we're saying there's a pair of chromosome that has two chromatids from the father. It has to be two chromatids. And a pair of chromosome that has two chromatids from the mother. This is what we call bivalent. Okay, so that's the difference. But bivalent chromosomes can also be considered homologous, if that makes sense to you. And they're considered homologous because they're coming, they have the same structure, but it's one's coming from the mother and one from the father. But what makes it bivalent is the fact that it's two chromosomes that have a total of 4 chromatids. Does that explain your point? Does that clarify your point? Yeah, okay, perfect. Does anyone have any other question? Does that mean, yeah, in metaphase, since we have 2 bivalent pairs, we have 8 chromatids. Let's go to metaphase. Sorry. No. No. Okay. If you look here, in metaphase, I understand why it confused you, but in metaphase, we have 1 bivalent pair, 2 chromosomes. So we have 1 chromosome from the mother. So this is, if you look at the picture, this is an X, okay? Or any more X, I mean, the shape X. And this is another shape X from the father. So if you count them, there is a total of 4 chromatids and 2 chromosomes. And what I meant to say, there are 2 chromosomes or 1 bivalent pair present at the metaphase. So there is 1 bivalent pair or 2 chromosomes present, 4 chromatids at a time. Okay, great. Any other questions? Guys, I understand that you've never taken the cell cycle in this much detail, so feel free to ask me about anything. It can be very confusing in the beginning. I understand that. So if you have any questions, feel free to ask. Okay. Since no one has any questions, I can proceed, but I just want to ask you, do you want to take a break or do you want me to just go with the other parts of the lecture? whatever you guys want, and I don't mind anything. So it's up to you. Would you like to have a break or should we? So 2 people said break and 1 said proceed. Can we have a prayer break? Yes, okay. So let's just have let's go back here at 8:30, a 7 minutes break. So you guys can pray, drink water, all of that, and inshallah we'll continue with the other parts of the lecture. So we're gonna have a 7 minutes break, okay? Okay, so now we're gonna move to the other parts of the lecture, which includes gametogenesis and in general. So before we get into it, we need to have a basic understanding of the anatomy of the female and the male's reproductive tract. So in the female's reproductive tract, we have these 2 white organs, which are called the ovaries. Now the ovaries, every month, release an egg in a process called ovulation. So they release an egg that will go all the way to the uterus. Now what's connecting these ovaries to the uterus itself is these 2 structures here that are called the fallopian tubes. So the eggs would be released from the ovary, it goes all over in the fallopian tubes, and then it goes to the uterus. Uterus is where implantation takes place, or basically implantation means where the fertilized egg would implement, or where pregnancy takes place. That's what it's trying to say. So that's just basic understanding of the female's reproductive organs. Now, when it comes to the male reproductive organs, if you look here, so we have the penis, and then here we have the scrotum, in which we have the testicles. Now, if we zoom in the testicles, it would look something like that. In the testicles, we have a lot of lobules. This is a lobule, and this is another lobule, and this is another lobule. Inside the lobules, we have a structure called the seminiferous tubules. So these tubules here in orange are called the seminiferous tubules. Now, what's important about these tubules is that this is where the sperm formation occurs. So the sperm is made inside the testicles, to be more specific, and the seminiferous tubules. After the sperms are made, they would go to this structure here, that's called the epididymis, or it's like yellow in this image. So the sperm would acquire motility in the epididymis, and it would be stored there until the time of ejaculation. Now, by the time of ejaculation, we have a structure called vas deferens. It would take the sperm and it will be ejaculated. Does that make sense? And it was just a basic understanding of how the anatomy happens, so that when we go to the information. Okay, perfect. So it makes sense. Now we're going to talk about the gametes, which are the hero of our lecture now. So we have the human egg or the ovum. This is a mature egg. So this is what a fully mature female egg looks like. And we're going to go into details of the structure surrounding it, inshallah, in the upcoming slides. And we have a mature sperm or spermatozoas. And this is the male sperm that's fully mature. So this is not how it starts initially. This is like the fully mature sperm, which is the one that we're used to, the one that has a tail and a midpiece. Inside the midpiece, we have a mitochondria. This is important. And then in the head, where we have the DNA, nucleus, and the acrosome. The acrosome is very important in the fertilization. And we're going to discuss how the sperm acquires all these features, inshallah, in the process of spermatogenesis. Does that make sense? Do you understand everything I've mentioned so far? Just very basic understanding, but I need you to understand them before we get into the more detailed explanation. Yes, okay, perfect. Now we're going to talk about gametogenesis. Now, if you break down the word gametogenesis, we have the word gametes. The gametes are either the sperm or the egg, and genesis means formation. So we're going to talk about the formation of either the sperm or the egg. Spermatogenesis is the formation of a sperm. Oogenesis is the formation of an ovum or the human egg. Okay? So we're going to start with spermatogenesis, and trust me, spermatogenesis is so easy. Yani, inshallah, you'll understand it really well. It's very, very easy to understand. Inshallah, once I explain it, you'll remember it. So one thing about males is that the formation of sperm starts at the time of puberty. So before puberty, males don't have any sperm in their body. Once they hit puberty, this is when like spermatogenesis starts to take place. And it takes place in the seminiferous tubules in the testes, the one I showed you earlier. And we said this is where sperm formation takes place. Okay? Now, the first sperm is what we call a spermatogonia. Spermatogonia for plural, spermatogonium for singular. So this is the first sperm. Now, this first sperm starts dividing by mitosis, and it's a diploid cell. Okay, so it's a 2N cell that keeps dividing by mitosis and produces a lot of spermatogonium. Now, after multiple divisions, the spermatogonium would differentiate into a spermatocyte. The process by which a spermatogonium becomes a spermatocyte is mitosis. So far, we didn't go into meiotic division. It's a mitotic division. It just keeps dividing like any cell, but then we change the name. We didn't do anything. We just changed the name of the cell. A spermatogonium divides by mitosis, and then we call it now a primary spermatocyte. Okay? So it divides by mitosis, forming the primary spermatocyte. Now, this primary spermatocyte, which is formed by mitosis, would then enter meiosis I. Okay? So we formed primary spermatocyte by mitosis. Now the primary spermatocyte would enter meiosis, and it would produce 2 secondary spermatocytes. So after meiosis I, we have secondary spermatocytes. It's just names so far. And the same process of meiosis that I explained earlier, now we're just having names for the cells. So we started with a spermatogonium that divided by mitosis. After the mitotic division, it produces a primary spermatocyte. It didn't change anything. It's just a different terminology. It's the same cell. Had to see it has the same color. Now, this primary spermatocyte would undergo meiosis I. After meiosis I, it would produce 2 secondary spermatocytes. Now, these 2 secondary spermatocytes would enter meiosis II, and they would produce 4 spermatids. Okay? So we started with spermatogonium, moving with mitosis to primary spermatocyte. This is all after puberty? Yes, it's all after puberty. Sperm formation does not begin in males before puberty. It starts at the time puberty. Spermatogenesis never starts before in males. Okay? This is important to remember, by the way. So after puberty, this all happens. Okay, of course. So spermatogonium, mitosis, we have primary spermatocytes. And then after it forms primary spermatocytes, it enters meiosis I. After meiosis I, we would have the secondary spermatocytes. Secondary spermatocytes would enter meiosis II, and then they would form spermatids. So far, do you understand everything? We're just going through meiosis, and we're having terminologies for the things. But you guys have to remember the terminologies. Yes, it makes sense. I'm glad. But make sure and you remember the terminologies, and remember, primary spermatocyte comes from mitosis, not meiosis. And secondary spermatocyte comes after meiosis I. Okay? Someone is asking me why aren't the secondary spermatocyte haploid? I knew someone would ask this question. Honestly, again, it's just a picture that I took from Google. It should be haploid. Like it should be because it's 23 chromosomes. So it is a haploid cell. So just ignore the picture. It's not specific, okay? Honestly, I put the pictures to make it easier to visualize, but the information that are written, I prefer if you take them from the slides themselves, okay? I'll try to remove any incorrect information, but honestly, when I was adding the pictures, I was mostly focused on the visualization part, okay? But inshallah, I'll correct them when I send you guys the slides. Okay. Now, since we've reached spermatids, I want you guys to notice one thing. It's very important. I know the spermatids are still circular in shape. Now, this is not the shape of sperm that we're used to. We're used to the tail, the head, the midpiece. This is how we study sperm in high school, صح? So how does a spermatid become the mature sperm that we know? This is another process that's called spermiogenesis. So we have spermatogenesis, which is the whole thing, and then we have spermiogenesis as a part. Now, if we go deeper in spermiogenesis, so spermatogenesis is this whole thing. Spermiogenesis is just part of spermatogenesis. So we had spermatogonium underwent mitosis, then we had the primary spermatocyte, and then we had meiosis I ended up with secondary spermatocytes, the meiosis II ended up with a spermatid, okay? Now we have 4 spermatids. These spermatids would then undergo spermiogenesis. Now in spermiogenesis, which is this process here, the sperm changes from the circular shape to this shape that we're used to, head, midpiece, and tail. So what is happening? Now the excess cytoplasm is shedding, which makes total sense. If you're talking about a circular cell and you compare it to the shape of the sperm, this one definitely has way more cytoplasm. So what's happening is that it's losing its excess cytoplasm. And there is formation of the tail, which was not there. And there is the acrosome formation. Remember the acrosome, which is present at the head, which I said is very important for fertilization. So this acrosome is formed in the process of spermiogenesis and DNA condensation. So it's no longer as like loose as it was in a circular cell. It's now more condensed in the nucleus, which is found in the head. Okay. So these are the main 4 things that are happening in spermiogenesis. Now, after we formed the sperm, we said the sperm would move to the epididymis. It's stored there until ejaculation. I've mentioned that when I was explaining the anatomy, and it's where the sperm gains motility. Okay? Does that make sense? Do you understand the process of spermatogenesis so far? Yes, perfect. Okay. Honestly, spermatogenesis is very easy. Inshallah, I know you guys won't struggle with it at all. Now we're going to move, if you guys understood spermiogenesis, to the question. So what is the correct sequence of cell stages in spermatogenesis? So almost all of you said D. We have almost 7 or 8 Ds, and that's the correct answer. Okay, I forgot to add the answer. No, I added, okay. It is this D, because we said we have spermatogonia undergoes mitosis, forming spermatocytes, primary, and then the primary undergoes meiosis I, forming secondary spermatocytes, which then undergoes meiosis II, forming spermatids, and then we'd have the spermatozoa, which was formed through the process of spermiogenesis. Someone's asking me, spermiogenesis occurs in the epididymis? No, it does not occur in the epididymis. It occurs in the seminiferous tubules. Because as I said, spermiogenesis is part of spermatogenesis. And spermatogenesis as a whole, it occurs in the seminiferous tubules. Epididymis is where the sperm is stored after it's formed. It's stored there and it acquires its motility in the epididymis, but it's not where spermiogenesis takes place. Okay, does anyone have any other questions? Where does the spermatogenesis occur again? So I'm just going to show you a picture. I said the seminiferous tubules, this one here, which are found in the testes of males, okay? These tubules here, the orange ones, this is the seminiferous tubules. This is where spermatogenesis takes place. And the testicles, particularly the seminiferous tubules. Of course. Does the sperm get mature in the epididymis? Primary sperm, okay. The sperm gets mature in the epididymis. It matures if by mature you mean it acquires its final shape, which is this one. Then no, it acquires its shape in the seminiferous tubules. But a part of maturation is acquiring motility, so it starts to move. The final maturation process by which it acquires motility happens in the epididymis. But the final shape of sperm, no, it occurs in the seminiferous tubules. The other question is primary spermatocyte to secondary is meiosis. Yes, it is meiosis, as you can see in this picture. From primary spermatocyte to secondary spermatocyte, it is meiosis I. Okay? Does anyone have any other questions? Of course. Is it clear so far? Yes, okay, perfect. Okay, so now we're gonna move to the other part. Oh, actually, I have another question. Okay. Can you guys answer this question? So we have B, 4B, B, B, B. Then yes, the answer is B, correct? Because spermatozoa is the mature sperm. And it makes total sense because we know also the outcome of meiosis is 4 cells, which happens in spermatogenesis. And we said we'd have 4 spermatids transforming into spermatozoa. So yes, the correct answer is 4. Now we're going to move to oogenesis, or the formation of eggs. Now, unlike males, for females, they start forming their eggs prior to being born. So all the eggs in the female's body are formed in the fetal life. Fetal life is basically when a female is still in the uterus, and you're still a fetus, and you're still in the uterus before birth. So all of our eggs as females, they're formed before we're born. And we don't make any new eggs after we're born. All of them are already made. And the females are born with their eggs. Okay? So the maturation of the ovum happens in the ovaries, which are the white organs that I showed you. And in each menstrual cycle, because we know females won't have menstrual cycles every month, in each menstrual cycle, one mature ovum will be released at the time of ovulation. Okay? Now, before I go into oogenesis, there is a very important concept that I want you to guys understand before I go into details, because this concept, I believe, is what confuses people the most. When we talk about oogenesis, we're talking about 2 simultaneous things that are happening at the same time. we're talking initially at the egg's development, okay? Which makes sense. This is what oogenesis is. But we're not only talking about the eggs, but we're also talking about the development of the follicle. So we have an egg. The egg is found inside a follicle. So we talk about the development of the egg separately, and we talk about the follicle. The follicle is basically the cells that are surrounding the egg separately. So when I say a primary oocyte or a primary egg or any name of the eggs, yani, it's different than saying a primary follicle. You need to understand from now that when I say primary oocyte, it's different than when I say a primary follicle. When I say a secondary oocyte, it doesn't mean no, it's in a secondary follicle. No, it doesn't mean this. Okay, do you understand this concept? And we have two simultaneous things happening together. And when I say primary oocyte, it's not the same as a primary follicle. They're two different things that are happening together. Okay, keep this concept in your mind before we go into oogenesis, because a lot of people confuse this part and know by thinking primary oocyte would mean a primary follicle. No, it doesn't. They're two different things. Okay, do you guys understand this concept? One said yes, but I just want to make sure that you guys all understand it, because this is very important. Like, this is probably the most confusing part of oogenesis. Okay, it's perfect. Okay. Alhamdulillah, since you all guys understand it, then okay, someone said, could you repeat? Sure, I will. So when it comes to studying oogenesis, we have the egg. Let's just throw the egg in red. So this is the egg, and the egg is surrounded by cells, okay? Let's say the green is the cells that are surrounding the cell. So excuse how I'm drawing it, it's just for the sake of understanding, okay? So these are the cells that are surrounding the egg. Now the egg can be called a primary oocyte, a secondary oocyte, whatever the name is. When I say a primary oocyte, it's different than when I refer to this whole structure as a whole. This whole structure as a whole is called a follicle, okay? So we have a follicle and we have an oocyte. And the follicle can be primary follicle, secondary follicle, and the oocyte can also be a primary oocyte, a secondary oocyte, okay? But when I say a primary oocyte, it doesn't necessarily mean that it's present in a primary or a secondary follicle. Like, they're not related. So a secondary oocyte, a primary oocyte can be present in a secondary follicle. That's what I'm trying to say. And when I say a primary oocyte, it doesn't mean that it's a primary follicle. No, it can be a primary oocyte and a secondary follicle, which I will explain more in details, but I want you to understand that these are two different things. Like, saying primary follicle is not equal to saying a primary oocyte. That's what I'm trying to explain. Do you understand this? And they're just two different things. I didn't explain what's happening so far. I just want you to understand that they're two different things. Okay? Do you understand it? Yes, thank you. Okay, of course. But, okay, let me just erase this. And yeah. So oogonium is the first developing egg, and it divides by mitosis, similar to the concept of spermatogonium. Again, oogonium, it's the first egg here, and it divides by mitosis, 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 until it forms a lot of cells. Now, by the third month of development, in the utero, some oogonia is the plural of oogonium, would start giving rise to what is known as a primary oocyte that enters the prophase of the first meiotic division. So we said we have meiosis I and meiosis II. So basically, this oogonium, it divides by mitosis, and then it's going to form a primary oocyte. Nothing hard so far. It's just mitotic division, and now we call it a primary oocyte. This primary oocyte would enter prophase 1 of meiosis I, and it would be arrested there. Now, it remains there until the time puberty. So as females, we're born with oogonium, okay? And then it enters the primary oocyte, after mitotic divisions, we would have the primary oocyte. And then this primary oocyte would be stuck in prophase 1 until the time of puberty. Now, what's keeping this primary oocyte stuck there is the release of oocyte maturation inhibitor by the follicular cells. Now, what are the follicular cells? I'll explain in the next lecture. Someone is asking me, can you please repeat? Sorry, in the next slide. Yes, sure, I will. So basically, the females, as we said, we're born with all our eggs. Females do not make new eggs after they're born. They're born with all the eggs. The egg is initially called an oogonium. Now, this oogonium keeps dividing by mitosis. So far, there is only mitosis happening. Now, the oogonium, after mitotic division, multiple mitotic divisions, we'd now call it a primary oocyte, which is still a diploid cell that's dividing by mitosis. Nothing happened so far. Okay, it's just similar to the spermatogonium becoming a primary spermatocyte. Now, this primary oocyte would enter meiosis I because it's a germ cell. It would enter meiosis I. But when it enters meiosis I, it does not complete meiosis I, just like sperm does. This doesn't happen in females. It would remain stuck in prophase 1 until the time puberty. So it's stuck there. And what's keeping it stuck is this oocyte maturation inhibitor. So it's, but as the name suggests, it prevents the oocyte from maturing and it's released by the follicular cells. What are the follicular cells? I'll explain in the next slide. But I just want you to understand, there are phases, there are pauses that are happening in the meiosis of oocytes. So it doesn't just go as sperm smoothly. No, it enters a phase, then it pauses, then it enters another phase, then it pauses again. The first pause is prophase 1. And the way I remember it is that we said the mnemonic is PMAS for prophase, metaphase, anaphase, telophase. So, but the primary oocyte is stuck in the first letter. So primary, first letter, so it's stuck in prophase 1. Does that make sense? So it's stuck in, okay, perfect. It's stuck in prophase 1. The oogonium is haploid, right? No, it's not haploid, it's a diploid cell. Okay, I'll explain a concept. So we have what is known as primordial germ cells. These primordial germ cells are diploid cells that can differentiate to either an oogonium or a spermatogonium. So initially the oogonium would be diploid, then it undergoes meiotic division to become a haploid cell. So initially, no, the oogonium is a diploid cell, it's not a haploid. But after meiosis, the ovum would end up being haploid. That's why it's undergoing meiosis, to reduce the number of chromosomes. Of course. Okay, so we agreed, and now we have a primary oocyte that is stuck in prophase 1. And what's keeping it stuck is this oocyte maturation inhibitor released by the follicular cells, okay? Now, we said we have the ovaries, and this is where this whole thing is happening in the ovaries. Now, from the epithelium of the ovaries, there are follicular cells that are developing. And it's surrounding the primary oocyte. So we said we have the primary oocyte that we just talked, and it's stuck in which phase? In prophase 1. Now, if you look at this picture here, you see this is the oocyte, this is primary oocyte, okay? And there are cells that are surrounding it, these blue cells. Do you see them? Now, these blue cells is what we call follicular cells. Where did these cells come from? It's just from the epithelium of the ovaries. They go down and they start surrounding the cells. When they surround the cells, we call them follicular cells. So the primary oocyte is now surrounded by follicular cells. Do you understand this? We have primary oocyte surrounded by follicular cells. Do you get this? Okay. Now, this whole structure where a primary oocyte is surrounded by follicular cells that are flat, it's called a primordial follicle. So one layer of follicular cells. Okay, when it's one layer of follicular cells and it's surrounding the primary oocyte, now we call it a primordial follicle. Keep in mind, primordial is different than primary. Primordial follicle is different than primary follicle. And a primary follicle is different than primary oocyte. Trust me, it's not hard. You just need to distinguish the terminologies. So when I say a primary oocyte, it's stuck in prophase 1, surrounded by a single layer of follicular cells. This whole structure is called a primordial follicle. Now, it remains here as in primary oocyte in prophase 1 until puberty. Someone is asking oocyte plus it's the oocyte plus the follicular cells, these follicular cells here, these blue cells, flat cells. Okay, this whole thing is called primordial follicle when it's a flat cell. Now, as the primary oocyte begins to grow, follicular cells change from these flat cells. So if you see here, these flat cells, okay, they would change to becoming a more cuboidal cells. They start proliferating. And when they are cuboidal and no longer flat, they're the same cells. Nothing changed. They're the same cells. They're just, instead of being flat, they start like proliferating and becoming more cuboidal. Now we call them granulosa cells. So we started with the flat follicular cells, F for F. When it's a flat, it's follicular. Then it matures a bit, the cells, not the ovum, not the oocyte. It's the cell that's surrounding it. The follicular cells would mature and they form more cuboidal cells. When they're forming cuboidal cells, now we call them granulosa cells. So it's just different names for the same thing. It was flat, we called it follicular, F for F, remember? And then the cells start to grow, becoming cuboidal. Now we call them granulosa cells. Okay. Now these granulosa cells that are surrounding the primary oocyte that is stuck in prophase 1 will form a primary follicle. Remember, when we were in follicular cells, it was primordial. Now when we have granulosa cells, now we call it primary. Does that make sense to you guys? And the oocyte is still a primary oocyte stuck at prophase 1. Okay, perfect. Now the oocyte is stuck because we said it's going to remain stuck until puberty. Perfect. Now the granulosa cells and the oocytes would start releasing glycoproteins. And as they're releasing glycoproteins, they would form this structure here in purple. I'm highlighting now. This structure here is called a zona pellucida. Someone is asking, this change happened at puberty? No, it's before puberty. At puberty, it's the primary oocyte that's changing more than the follicle itself. Okay. I'll explain what happens in puberty more when we get to the continuation of the meiosis, of course. So we had follicular cells in the primordial follicle, and then the cells would kind of enlarge, become cuboidal. Now we call it a granulosa cells. And now when it changed from follicular to granulosa, the follicle is now, instead of being called primordial, we call it primary. The oocyte is still a primary oocyte stuck at prophase 1. Okay? And the oocytes and the granulosa cells release glycoproteins that would form the zona pellucida. Now the zona pellucida, it's not significant in this lecture. However, it is very significant in your fertilization lectures, because zona pellucida, it's going to remain surrounding the egg, and the sperm has to penetrate it. That's basically why I'm explaining the zona pellucida for you, so that when you go to your fertilization lectures, you know where did the structure come from. It's basically glycoproteins that are released from the ovum and from the granulosa cells, and it's going to form this layer of glycoprotein that is called the zona pellucida. But now we're not going to focus on it. I just want you to know that it exists and that it's a layer of glycoprotein released from granulosa cells and the oocyte itself. Now we're going to focus on the follicles themselves. So we had a primordial follicle, primary oocytes, like a prophase 1 with follicular cells. I'm going to keep repeating it so you guys, inshallah, try to memorize it with me. And then the follicular cells would enlarge, cuboidal cells. Now we call them granulosa cells. They're the same cells, just different sizes, basically. And the oocyte is still a primary oocyte stuck at prophase 1. And now instead of calling it a primordial follicle, now we call it a primary follicle. Okay, does that make sense so far? I'm going to get to the secondary follicle in a bit. When is this development of the follicle happening? So this development of the follicle, okay, I'll just explain briefly what's happening. The female is born with a primary oocyte stuck at prophase 1, as I said. So this is the oocyte. Now when the follicle, each month, basically, a follicle would begin to mature. So each month, they're a group, actually, to be more specific, at each month, around 15 to 20 follicles begin to mature, and only one follicle reaches the full maturation stage, and it's basically the one that's released during ovulation. Does that make sense? So we have them stuck. You got it? Okay, perfect. So we have them stuck at prophase 1 for the oocyte. The follicles, at every month when in the menstrual cycle, this process happens. 15 to 20 follicles start to mature, and only one's going to be released. The follicles divide by mitotic division, so the layers grow. Yes. Basically, yeah. Because if you look here, now we were in like follicular cells. Now in the secondary follicle, you'd have way more cells, and I'm going to explain it in more details. We started with follicular cells, then granulosa cells, and then here in the secondary follicle, what you would notice is that we'd start to have spaces here that are fluid-filled. When you start, I'm sorry, the arrows would be in the opposite direction, but anyway. So when you have these fluid-filled spaces appearing between the cells, this is when we start calling it the secondary follicle. Now, so far, the oocyte is still a primary oocyte stuck at prophase 1. Someone is asking me, can you repeat? Sure, I'll explain the secondary follicle, then I'll repeat the whole thing. At which stage is follicle end before puberty? It's at the primordial follicle stage. So this is what the females are born with. And each menstrual cycle, the 15 to 20 of the follicles begin to mature, okay? And for the repeating, inshallah, I'll explain the whole thing again. But now I'm going to give you an idea. When ovulation happens, are they still stuck in metaphase of mitosis 2? I didn't go to metaphase of mitosis 2 yet. I didn't go to that part at all. We're still at prophase 1. Prophase of the first meiotic division. I didn't mention anything about metaphase so far. I'll explain it later on, but I don't want to confuse you. I'm going to go step by step, okay? When I go there, inshallah, I'll explain your points. But I don't want to confuse you so far. Okay. So when you start seeing these fluid-filled spaces in between the follicular cells, now we call it a secondary follicle. So that's the hallmark. The oocyte is still a primary oocyte stuck at prophase 1. So far, no ovulation has happened, nothing. The follicles are just maturing, okay? Now, when the secondary follicle has matured, there is a hormone in the female body that, actually even in males, but there is a hormone that's called luteinizing hormone. It's the hormone that's responsible for ovulation. So at the time of ovulation, at each menstrual cycle, this hormone is released. So this surge in LH at every menstrual cycle is what causes the completion of meiosis I. So it's basically, it removes the inhibition that was happening. So it's no longer stuck at prophase I. It continues the first meiotic division. And now we would have the secondary oocytes. Okay? So we'd have the secondary oocyte. Now, after we form the secondary follicle. Now the cells then enter meiosis II, but are arrested in metaphase II, يعني basically in metaphase of the second meiotic division. Now, if you look here at this picture, so we started with oogonium, which I've mentioned, and then now I'm going to talk about the oocyte. I'm not going to mention anything about the follicles. I'll associate it with the follicles later on. Now we'll just focus on the oocyte. I'm going to repeat from the beginning. So we had oogonium, which we said is a diploid cell. It starts dividing by mitosis. Now, after multiple mitotic division, we start to call it a primary oocyte. But this primary oocyte is stuck in prophase I. Now, why is it stuck in prophase I? Because of the release of oocyte maturation inhibitor. Okay? It remains stuck until puberty. Because females before puberty, they don't have ovulation, so basically it doesn't mature. Makes total sense, صح؟ We're just talking about the oocyte. Don't overwhelm yourself with the follicle for now. Just understand that it's stuck until puberty. Now, by the time of puberty, what happens is, now I'm going to come to the follicles a bit. In every menstrual cycle, we have the phase of ovulation. And ovulation means basically an egg is being released. So, at the time of ovulation, 15 to 20 of the follicles begin to mature. But only one wins this race and reaches the full mature stage. And it's the one that's ovulated. Now, what causes this ovulation is a surge in a hormone called LH. When there's a surge in this luteinizing hormone or LH hormone, meiosis I is completed, because it will finish meiosis I. It would enter meiosis II, but it would be stuck again at the second metaphase. So it doesn't continue meiosis II. Another important point to mention when we're talking about oogenesis is that unlike spermatogenesis, the cytoplasm distribution between the cells is not equal. So if you look here at this part of the picture, you're going to see we formed a secondary oocyte. So this part took the most of the cytoplasm. And because very little cytoplasm went to the other one, we're going to form a polar body. Now you might ask me, what is a polar body? It's basically a structure that will eventually degenerate because it has very little cytoplasm. So the genetic material, the cytoplasm, and everything would mostly go to the secondary oocyte. The polar body would eventually degenerate because it doesn't have enough cytoplasm for it to survive. So we'd end up with only one oocyte. The second would go. Okay, that's the difference in meiosis and oogenesis and meiosis and spermatogenesis. Okay, there's a very good question. A girl asked, does secondary follicle form first before the secondary oocyte? Yes, that's very true. Because when it comes to the follicles, now I'll focus on the follicles. Girls are born with primordial follicles, which is a primary oocyte stuck at prophase I, and it's surrounded by flat follicular cells. At every menstrual cycle, the follicle begins to mature. These follicular cells will become granulosa cells. Okay, it's because they're larger. It's the same cells. They're just cuboidal rather than flat, so we call them granulosa cells. And now instead of calling it a primordial follicle, we would call it a primary follicle. The oocyte is still a primary oocyte stuck at prophase I. When we go to the secondary follicle, it's still a primary oocyte stuck at prophase I, but the only difference is that we start to have fluid-filled spaces appearing between those cells or the granulosa cells that were surrounding the oocyte, okay? So at secondary follicle, it's still a primary oocyte. Then what happens is there's a surge in luteinizing hormone, or LH, which basically causes the ovulation to take place. When this surge happens, the primary oocyte continues meiosis I, so it would end up with a secondary oocyte, and it's only one secondary oocyte. Why? Because the second part is now a polar body, and we said it's going to degenerate. Plus, we don't care about the polar body because we don't have it anymore because it has very little cytoplasm that it does not allow it to survive. Now, after that, when there is a surge and we form the secondary oocyte, it's these fluid-filled spaces would start to join together. And when they join together, they would form this very large space, and we call the space the antrum. When we have this antrum, or very large fluid-filled space, this is called a Graafian follicle. And the oocyte here is a secondary oocyte stuck at metaphase II. That's the difference. In secondary follicle, it is still a primary oocyte stuck at prophase I. Then the surge of LH happens. It allows the primary oocyte to continue meiosis I. It's no longer stuck at prophase of metaphase I. So it becomes a secondary oocyte. Also, by the time the surge of LH happens, these fluid-filled spaces in the secondary follicle join. They form a very big space called the antrum. Now, when this antrum is formed, we call it a Graafian follicle. It's also referred to sometimes as the antral follicle because of the name of the space. Okay. Now, another important point, and when the cell enters meiosis II, the secondary oocyte, it will again be stuck at metaphase II, and it will remain stuck at metaphase II until the time fertilization. So a female basically will never have her eggs fully matured until the time fertilization. Okay? So it remains stuck at metaphase of the second meiotic division. And the way to remember it, again, I go back to this PMAT, prophase, metaphase, anaphase, telophase. We said in the primary oocyte is stuck at prophase 1. So all 1, 1, 1. The first letter, the first part of meiosis, so prophase 1, and the primary oocyte, all ones. Now, the secondary oocyte, which is the number 2, will go to the letter 2, M for metaphase, and it's the secondary meiotic division. So this is the best way to remember it, in my opinion. So just remember the mnemonic PMAT. It's not really a mnemonic, it's just an acronym. But anyway, metaphase, metaphase is the second letter in PMAT. So a secondary oocyte would be stuck in metaphase 2. So all 2s, if that makes sense. Yeah. This is how I remembered that when I was in first year. Each month, a new Graafian follicle is formed and stuck in metaphase 2. Yes, exactly. Each month, when there's a menstrual cycle, it's the time of ovulation, the Graafian follicle is formed. Someone is asking, does secondary follicle contain secondary oocyte? No. Secondary follicle contains a primary oocyte. And I have it written for you guys here so that it doesn't confuse you. Let's go back here. So we said the primordial follicle, which is this one basically, which is surrounded by follicular cells, is a primary oocyte with one layer of follicular cells surrounding it, and it's arrested in prophase 1. Then the primary follicle is still a primary oocyte, but instead of having follicular cells, now we have the cuboidal granulosa cells, and it's still arrested in prophase 1. Now the secondary follicle is a primary oocyte arrested in prophase 1, but between the granulosa cells, now we have spaces. Remember here, we said fluid-filled spaces begin to appear between the granulosa cells. But the oocyte is still a primary oocyte stuck in prophase 1. Now the surge of LH happens, which means ovulation has happened. But the primary oocyte continues meiosis I, and after it continues meiosis I, now refer to it as the secondary oocyte. By the time it's happening, and because we said two simultaneous things are happening at the same time, by the time the primary oocyte becomes a secondary oocyte, these fluid-filled spaces would join together, they would form an antrum. Now we have a Graafian follicle. So the secondary oocyte stuck at metaphase 2 is only present at the Graafian follicle, as I've written here. So Graafian follicles is a secondary oocyte surrounded by coronary data. I'll explain what coronary data is. It's just part of the cells. The same cells, just different names. And there is an antrum there. And it's stuck at metaphase 2. I'll add this to your slides. It's stuck at metaphase 2. Okay? Secondary oocyte stuck at metaphase 2 surrounded by coronary data. What's coronary data? Let's understand what it is. So we said out of 15 to 20 follicles maturing each menstrual cycle, only one would reach the Graafian follicle stage, which is the fully mature stage that we just talked about. Okay? Now the layer of granulosa cells covering the oocytes are now termed coronary data. You see the antrum keeps getting bigger and bigger and bigger. Now these cells here, which are the granulosa cells, the same cells would remain called the granulosa cells. But these cells here that are immediately on top of the egg are called coronary data. Now, coronary data basically means a radiating crown. So the way for you to remember, it's like a crown on top of the eggs. That's why they're the same cells. They were follicular, then granulosa. We now call them coronary data cells. Okay? So it's just the same cells, just different names. So follicular, granulosa, coronary data. Now the ovaries release the Graafian follicle. Now what is a Graafian follicle? It's a secondary oocyte stuck at metaphase 2 with coronary data surrounding it. So this is like the whole thing is released. The released ovum would be picked up by the fallopian tubes. Remember what the fallopian tubes were? We said they're the ones that are connecting the ovum to the uterus. So the egg would be released. This is the Graafian follicle with coronary data. And then it would move from the fallopian tubes all the way to the uterus. So this is how it's happening. This whole thing is happening in the ovaries. Okay. And then fertilization with sperm alleviates meiosis II arrest. So without fertilization, the eggs are never fully mature. Thus, technically, the female gametes only truly becomes haploid. So whenever we say truly haploid, yani the chromatids are 23 and the chromosomes are 23, only after fertilization. Does that make sense? I'm going to repeat it one more time with details, but I just want you to tell me if you understood everything. If you have something that you didn't understand, so I can focus on it when I'm repeating. Do you guys understand everything so far? I know oogenesis can be a bit harder. Yes, okay, I'm glad. Alhamdulillah. Does anyone have any specific question? Someone is asking, where is the zona pellucida in the graphene follicle picture? The zona pellucida is still there. Actually, if you look here, this is the graphene follicle. This is the egg. And this red thing here surrounding it. This is the zona pellucida. And on top of the zona pellucida, we have the corona radiata. That makes sense to you. So in the graphene follicle stage, it's diploid. It's not diploid, it's haploid because it has 23 chromosomes, but it has 46 chromatids. So it is haploid. It is haploid, but not, when they said fully truly haploid, they mean like the chromosomes and the chromatids are both 23. I understand that it can be a bit confusing, but I'm gonna repeat the whole thing. Actually, I'm just gonna, how 3 polar bodies form at the end from 1? Okay, this is a good question. Someone is asking me how do we have 3 polar bodies at the end? I'm just gonna erase the mess that I drew, so it makes way more sense. This goes back to our meiosis explanation. Remember, we said the end of meiosis, because we have meiosis I and meiosis II. So we had 1 cell, and then the cell divides. After meiosis I, we'd end up with 2 cells. And after meiosis II, we'd end up with 4 cells. Just like sperms, we ended with 4 sperms and everything. Now, what happens in oogenesis is that the cytoplasm distribution is not equal. So in the first meiotic division, this is the first meiosis, okay? In the first meiotic division, 1 of these cells would become a primary oocyte. After meiosis one would become a secondary oocyte, and the other one would become a polar body. So basically, it degenerates because it doesn't have enough cytoplasm, so it doesn't survive. So we're done. This is the first polar body. It's gone. Plus, we don't have it anymore. Now, what's remaining is this polar body would eventually divide, and it's going to form 2 polar bodies. It's just a polar body, and it's forming more polar bodies. Eventually, all of them will degenerate, so we don't really care. The polar bodies would all be gone. Now we have this secondary oocyte. So it would divide by meiosis II. One will turn into an ovum, and the other one would turn again into the polar body because it doesn't, basically, it doesn't undergo, it doesn't have enough cytoplasm for it to survive. Sorry, I was reading another question. I'll answer the other question after I explain this. So it doesn't undergo, it doesn't have enough cytoplasm, so it doesn't survive. So moral of what I'm saying is that after meiosis I, 2 cells would be the product. In this case, there's no enough there's not equal cytoplasm distribution, so 1 would become a polar body and it will disintegrate in the end. But 1 will become a secondary oocyte. Now we undergo meiosis II, so even the polar body would divide in meiosis II because there is still a division. The polar bodies would form 2 polar bodies, which we call secondary polar body, but we don't really care because both of them will eventually degenerate, so we don't really care. After meiosis II, we'd have an ovum, which is this one, and another polar body because not enough cytoplasm going there. We said cytoplasm distribution is not equal, so we'd end up with 3 polar bodies and 1 ovum. And this is because we go through 2 meiotic divisions, so meiosis I and meiosis II. Do you understand this? If you don't, it's fine, I can repeat. The other question is, when follicles mature during ovulation, they start at the primordial follicle and end as graphene follicle. Exactly. So primordial to primary to secondary to graphene, and then the graphene is the one that's released with the corona radiata. Of course. Does anyone have any other questions? Guys, I understand that oogenesis is not the easiest to understand. Where does the zona pellucida go? The zona pellucida is not gone. It's released in the ovulation process. So in the ovulation, basically what is released is the egg, and it's surrounded by, so this is what's released during ovulation. The egg, surrounded by zona pellucida, it's still released in the ovulation, and with the corona radiata cells, which I'm going to represent in the color green. okay? So the egg is the one in like dark blue, and then in red is the zona pellucida, and then the corona radiata is the one in green. And when you go through your fertilization lectures, you're going to see that the sperm has to penetrate all these three layers. It has to penetrate the corona radiata, which are these cells, the zona pellucida, and the like layer surrounding the ovum itself. So all three of them will be released during ovulation. That's if fertilization takes place, the sperm has to penetrate all of these. Now, if fertilization does not take place, this whole structure basically would become a corpus luteum. Oh, sorry, excuse me. So this structure will be released. I mean, the remaining of this structure will become the corpus luteum. So we release the egg, and then corpus luteum is the remaining of this. Okay, so the corpus luteum is everything minus what we released during ovulation, which was the egg, the zona pellucida, and the corona radiata. The remaining structure will become something that's called corpus luteum. Now, corpus luteum, I'm not really sure if you should know it at your level, but I'm just going to explain it for the sake of understanding. It is important in case of pregnancy, like if fertilization takes place, the corpus luteum releases an important hormone. This is not for you to study. I'm just saying so you understand what's happening. Release a hormone called progesterone, which basically helps the pregnancy to stay there. If pregnancy does not take place, then corpus luteum would degenerate. So this is why corpus luteum is formed. And it's like the remaining of the whole thing. And if we take this structure, this image at the top, and if we release this during ovulation, once this structure is released, everything that's remaining will form the corpus luteum. This will be released. And what do we see here? We see the egg, the zona pellucida, and the corona radiata. They're all released during ovulation. So for fertilization, our secondary oocyte is the one that's being released. Yes, so secondary oocyte is the one. Yeah, secondary oocyte is the one that's being released, and then it does not complete meiosis II, so it doesn't go from secondary oocyte to a mature ovum unless fertilization takes place. Exactly. Yes. I'm going to repeat it one last time, everything smoothly together. And if anyone has any questions, you can always ask. So someone is asking, metaphase continues before or after the ovum is released to the fallopian tubes? No, metaphase continues before. Metaphase continues before because the surge in LH allows the primary oocytes to continue meiosis I. Now it goes to meiosis II, and it's stuck at metaphase II. And then we form the Graafian follicle. This horse structure is released to the fallopian tubes as a Graafian follicle, and a secondary oocyte stuck at metaphase II before being released at the fallopian tube. So when it's released, everything's done. What's the function of corona radiata? Corona radiata is, you see those cells that were here, that were surrounding it? It's the one that initially, it's one of the cells that initially released the glycoprotein and formed the zona pellucida, but it's very important in the fertilization. Why is that? Because I don't know if you know this from your high school, but in fertilization, so many sperm try to penetrate and so many sperm try to fertilize the egg. And only one sperm should be able to like fertilize. The function of corona radiata and zona pellucida is that it protects the cell, the ovum itself. It does not allow something that's called polyspermy to happen. So it does not allow more than one sperm to be able to fertilize, because it's only the very strong sperm will be able to penetrate the corona radiata and the zona pellucida to enter inside the ovum. So it's kind of a protection for the egg from having like multiple sperm and like, yeah, and like degenerating basically. So it protects the egg. That's the purpose of corona radiata. Other than the fact that it releases glycoproteins and forms zona pellucida. Does that make sense to you? But always remember that corona radiata is the same cells, like follicular, then granulosa, then corona radiata, they're all the same cells. Just different stages, have different names. Okay. So I'll explain the whole thing here at the checkpoint. So we have on one side, I'll explain the eggs, and on one side, or the oocytes, I will explain the follicles. So initially, we'd have the oogonium, okay? This oogonium would undergo mitosis. Just write mitosis. Mitosis, okay? I'm not going to continue, but let's just remember that this is mitosis. When it undergoes mitosis, this oogonium would form what is known as a primary oocyte, okay? I hope this makes sense, right? This primary oocyte, after mitosis, will then enter the first meiotic division, which makes sense because it's a germ cell, so it divides by meiosis. Now, in meiosis I, the oocyte will not be able to continue. It would be stuck at which phase? Can someone tell me at which phase of meiosis it would be stuck at this point? Prophase one, prophase one, prophase. Exactly. So all of you said prophase, and it's the correct answer. So here it would be stuck at prophase one. And it would remain stuck here until ovulation takes place, which happens at the time puberty. Because before puberty, females do not have the ovulation process. This does not occur in female until puberty. So by the time puberty, at every month of the menstrual cycle, there's a stage of ovulation where an egg is released. So every month, around 15 to 20 of these eggs begin to mature. And what allows this maturation is the surge in LH. So if they ask you what's like removing this inhibition and allowing it to proceed from prophase to like to the second, and it continues basically meiosis one, it's the surge in LH hormone, because this surge is required for ovulation. Now, whenever we have the surge in LH or the luteinizing hormone, the primary oocyte would enter the second meiotic division. Okay, it will form the secondary oocytes and a polar body. And we said it forms a polar body because the distribution of the cytoplasm is not equal. So most of the cytoplasm would go here, and very little cytoplasm would go here, forming a polar body, and then this will eventually degenerate. Okay? Now, the secondary oocyte, can someone tell me where would it be stuck? Metaphase 2. Metaphase. Another metaphase? Exactly. So all of you said metaphase. Metaphase 2, yes, exactly. All of you said metaphase, and it's the correct answer. The secondary oocyte would be stuck in metaphase 2 until fertilization takes place. So if the ovum is not fertilized, it's never really fully mature. Now, let's say fertilization did take place and it allowed meiosis 2 to continue. The polar bodies would again divide by meiosis 2, and they would form two other polar bodies, which will eventually degenerate. And the secondary oocyte would then form a mature ovum and a polar body. Now, the polar bodies, all of them, let's say in yellow, all of them will degenerate. The ovum is now mature and it's ready to continue like maturing. It's the mature ovum, basically. Now let's talk about the follicle. So this is the oocyte. Let's talk about the follicles. Females are born with a primordial follicles. Now, what is a primordial follicle? It's basically an egg surrounded by flat follicular cells. Okay, so these are the follicular cells that are surrounding the oocyte. So this is a primordial follicle. Every month during ovulation, these follicles begin to mature. Now we'd have an egg, and rather than having flat cells, we would now have the same cells. They would mature more, and they would form cuboidal cells. Can someone tell me what's the name of the cuboidal cells I'm drawing right now? They're the same cells, they just have a different name. Does anyone remember what the name is? Granulosa cells, granulosa, exactly. Granulosa. Someone said follicular cells. They are the follicular cells, but when they mature, we no longer call them follicular. When they're flat, remember F for F, when they're flat, we call them follicular. When they're more cuboidal, we call them granulosa cells. But yani, I understand your point. They are the same cells, but we just have different terminologies for them. Now, these granulosa cells would release a layer of glycoprotein, and it's going to start around between the eggs and the granulosa cells, this red structure here, which we call the zona pellucida, which matters in fertilization. So you're going to understand it more in your fertilization lecture. After that, these granulosa cells continue to divide, and there's a lot of proliferation. And now, let's just draw a lot of them. Just draw a lot of granulosa cells, okay? And an egg, and imagine that there's a zona pellucida. Zona pellucida is still there. I'm just not gonna draw it. I'm gonna take an eraser and just gonna, oh no. I didn't mean to erase the whole thing. I just meant to make like fluid-filled spaces. When we have fluid-filled spaces, we call it a secondary follicle. That's what I'm trying to illustrate here. So now, when these fluid-filled spaces join together and they form a very big space, now we call it this very big space, and like there's an egg here, we call it a Graafian follicle, okay? So now, I'll ask you guys questions, and I want you to focus. So now we said we have two, one of two. We either have a primary oocyte stuck at prophase 1 or a secondary oocyte stuck at metaphase 2. So can someone tell me, in primordial follicle, which one would you have? Is it prophase 1 or metaphase 2? Prophase 1, exactly. So it is a primary oocyte stuck at prophase 1. برافو. طيب. In the primary follicle, is it a primary oocyte? Is it a primary oocyte stuck at prophase 1, or is it a secondary oocyte stuck at metaphase 2? It's a primary oocyte stuck at prophase 1. Yes, exactly. برافو. طيب. At the secondary follicle, where we have fluid-filled spaces, but they're not joined together, is it a primary oocyte stuck at prophase 1, or is it a secondary oocyte stuck at metaphase 2 in the secondary follicle? Bravo. I'm just gonna wait for more people to answer. I want you to guys think about it. Yes. Okay, all of you answered it correctly. Bravo. So it is, yes, it is a prophase, a primary oocyte stuck at prophase 1. طب at the Graafian follicle, is it a primary oocyte stuck at prophase 1, or is it a secondary oocyte stuck at metaphase 2? Yes, bravo, all of you. Alhamdulillah, bravo, all of you answered it correctly. It is a secondary oocyte stuck at metaphase 2. طب can someone tell me, actually I have this as a question later on. خلاص, I'll ask you later because I have the questions. But does anyone have any questions about anything I've explained in oogenesis? Like, do you understand everything I've explained so far? I can understand that it could be confusing, but I tried my best to simplify it, really. But if you have any questions, I'm more than happy to repeat it. You cleared everything. Thank you so much. Oh, I'm glad. Of course. Does anyone have any questions? Okay. But since you guys don't have questions, I do have a couple of questions for you. So can you all answer this? So all of you said the same answer, which is D, and the answer is correct. Oh, I'm glad all of you answered this correctly, and I'm genuinely happy. Yes, it is D. It is metaphase of meiosis II, but if it was a primary oocyte, then the answer would be A, and it would be prophase of meiosis I. Bravo. طيب. Which of the following has an antrum? Okay, so all of you said C, and it's the correct answer. Remember, the antrum is this very big space where the fluid-filled spaces begin to join, and it's present in the Graafian follicle, and aslan, another name for a Graafian follicle is an antral follicle. So this is where the antrum is present. Okay. So this is just a final comparison. A sperm produces an uninterrupted sequence, whereas oogenesis has very long pauses. We said, there is prophase 1 and metaphase 2. Spermatogonia continually self-replenish, whereas oogonia are limited in supply because spermatogonia, males, we said they start also forming them after puberty and they continue forming them for life. Whereas oogonia, we're born with all our eggs, so we have limited supplies of them. One oogonium results in one daughter oocyte because we said we form 3 polar bodies and 1 oocyte, whereas 1 spermatogonium results in 4 daughter sperms. Spermatogenesis starts at puberty, whereas oogenesis starts before birth. These are the main differences in spermatogenesis and oogenesis. If you have any questions, or if you have any point that you didn't understand, or if there's any point in your lecture that I didn't include in my slides, feel free to email me. Inshallah, I'll be answering all your questions. And I hope you benefited. Someone said, yesterday I had a panic attack from not understanding embryo, but you made me happy. Thank you so much. This is the sweetest thing ever. Thank you. This literally made my day. I'm glad I helped. Where will the PowerPoint be uploaded? Inshallah, your BLCs will send it to your group. Yani inshallah, in your announcement group, the PowerPoint slides will be sent. Of course, I'm glad I helped you guys. Oh, of course. Someone said, could you please go back to the slide with oogenesis overview? Sure. This one? Yes. Sure. Do you have any questions here? Let me just so it's clear. All clear. Okay. Okay, thank you. I hope you guys do amazing in your final. And don't forget, you can always email me if you have any questions. إن شاء الله يا رب تكون it benefited you. Okay, thank you guys. Have a good day. Bye bye.